Before written language, before cities, before agriculture, before almost everything recognizable as civilization, humans and their ancestors lived through millions of years that shaped every aspect of who we became. The Stone Age alone stretched across 2.5 million years, a timeline so vast that recorded history feels like a blink by comparison. During this immense span, ancient toolmakers transported rocks across distances exceeding 60 miles to craft the perfect blade. Neanderthals painted symbolic art on cave walls 65,000 years ago, and giant ground sloths standing 12 feet tall roamed continents alongside early humans. Recent genetic discoveries revealed in 2025 show that modern humanity didn't evolve from a single lineage, but from at least two ancestral populations that diverged 1.5 million years ago and later merged. Prehistoric history contains countless revelations that challenge assumptions, inspire wonder, and illuminate the deep roots of human ingenuity. Settle in for a journey spanning millions of years, filled with discoveries that rewrote what scientists thought they knew. In July 2011, archaeologists Sonia Harmond and Jason Lewis made a discovery that would redefine humanity's technological origins, not through meticulous planning, but through a simple wrong turn. Their team had been traveling through the hot, arid landscape near Lake Turkana in Kenya, heading to a known fossil site, when they took an incorrect route and found themselves in uncharted terrain. Deciding to survey the unfamiliar area, they spotted stone artifacts scattered across the ground. What they had stumbled upon was Lomekwi 3, an archaeological site harboring stone tools that dated back 3.3 million years, pushing the known archaeological record back by a staggering 700,000 years. This discovery fundamentally challenged the prevailing narrative that toolmaking began with the genus Homo, revealing instead that even more ancient hominins possessed the cognitive sophistication to deliberately shape stone into functional implements. The tools themselves were substantial, with the largest specimen weighing 15 kilograms, likely used as an anvil for battering and processing materials. Around 20 well-preserved artifacts were excavated from undisturbed sediment layers, with an additional 130 found on the surface, including cores, flakes, and hammers. In one telling instance, researchers matched a flake to its parent core, demonstrating that a hominin had napped the tool and discarded it at that very location millions of years ago. The toolmakers at Lomekwi exhibited intentional selectivity, purposefully choosing large, heavy blocks of high-quality stone while ignoring smaller pieces of the same material available nearby. Analysis revealed that the cores had been rotated as flakes were struck off, indicating an understanding of fracture mechanics and deliberate technique rather than random percussion. Archaeologist Alison Brooks confirmed that the tools could not have been created by natural forces, and the dating evidence, anchored to volcanic ash layers and magnetic reversals in the sediment, was fairly solid. The implications of the Lomekwi discovery extended far beyond pushing back dates on a timeline. For decades, conventional wisdom held that stone tool production emerged with the genus Homo around 2.5 to 2.6 million years ago, coinciding with climate shifts and the expansion of savanna grasslands in Africa. The Oldowan tool industry, named after Tanzania's Olduvai Gorge, where such tools were first identified in large quantities, represented what scientists believed was the earliest chapter of technological innovation. Homo habilis, dubbed handyman when it was described in 1964 by Louis Leakey, Philip Tobias, and John Napier, was long considered the first toolmaker, a species with a slightly larger brain case than Australopithecus, and the cognitive capacity to modify stone deliberately. But Lomekwi's tools predated not only Homo habilis, but the entire Homo genus by 500,000 years. The species responsible remains uncertain, though candidates include Australopithecus or Kenyanthropus platyops, a hominin whose fossils were found in the same region. Harmon proposed that these artifacts represent a distinct technological tradition she named Lomekwian, separate from the later Oldowan industry. Whether the Lomequian technology was forgotten and later reinvented, or whether it formed a continuous thread leading to Homo's innovations, remains an open question. What is clear is that tool-making capability emerged much earlier in the hominin lineage than anyone had anticipated, reshaping models of cognitive evolution. The older one tradition that emerged 2.6 million years ago represented a refinement and systematization of earlier tool-making practices, though it shared fundamental similarities with the Lomequian techniques. Named after Olduvai Gorge in Tanzania, where Louis and Mary Leakey first identified these implements in large numbers during the 1930s, Oldowan tools became the longest-lasting technological tradition in human history, persisting for approximately 700,000 years, with remarkable consistency across vast geographical ranges. The basic Oldowan toolkit consisted of several standardized forms: choppers with a single sharpened edge, created by removing flakes from one side of a stone core, scrapers fashioned from flakes with deliberately retouched edges, and hammerstones used to strike flakes from cores. These tools were not ornate or complex by modern standards, but they represented a cognitive watershed, the ability to envision a desired end product, select appropriate raw materials, and execute a sequence of precise strikes to achieve that vision. Archaeological sites from this period reveal that toolmakers often transported high-quality stone over considerable distances, sometimes exceeding 10 kilometers, demonstrating both geological knowledge and advanced planning. At Olduvai Gorge itself, researchers found that hominins preferentially selected fine-grained volcanic rocks like basalt and quartzite, while bypassing readily available but lower-quality materials. This selectivity wasn't random. The chosen stones fractured predictably when struck, producing sharp-edged flakes ideal for cutting, while inferior stones shattered unpredictably or produced dull edges. The cognitive demands of stone tool production have been rigorously studied through experimental archaeology, where modern researchers attempt to replicate ancient techniques. These experiments reveal that even simple Oldowan tools require surprisingly sophisticated mental capabilities. A toolmaker must understand that striking a stone at specific angles with controlled force will detach a flake, leaving both a sharp-edged flake and a core suitable for further reduction. This understanding involves grasping abstract principles of conchoidal fracture, the curved, shell-like patterns that form when certain stones break, and applying those principles through precise motor control. Neuroscientific research using brain imaging has shown that modern humans replicating Oldowan tool production activate brain regions associated with complex motor sequencing, spatial reasoning, and hierarchical planning. These are the same neural networks involved in language processing, leading some researchers to propose that the cognitive foundations for both tool-making and linguistic communication may have co-evolved. Furthermore, archaeological evidence suggests that tool-making knowledge was transmitted socially across generations, implying teaching, learning, and possibly even rudimentary pedagogical strategies. Sites where hundreds or thousands of stone flakes and cores cluster together, known as lithic scatters, indicate that multiple individuals repeatedly return to favored napping locations, perhaps because experienced toolmakers instructed novices there, or because the sites offered access to water, shade, and quality stones simultaneously. The practical applications of these early stone tools extended far beyond what their crude appearance might suggest. Sharp stone flakes could slice through animal hide more efficiently than human fingernails or teeth ever could, providing access to protein-rich meat and calorie-dense fat. Hammerstones could crack open large bones to extract marrow, a nutrient-dense food source that carnivores with powerful jaws like hyenas typically monopolized. Choppers and scrapers could process tough plant materials, roots, tubers, seeds, making them more digestible and releasing nutrients that would otherwise pass through the digestive system unabsorbed. In effect, stone tools functioned as external digestive organs, pre-processing foods before consumption, and dramatically expanding the range of edible resources available to early hominins. Rick Potts, a paleoanthropologist at the Smithsonian Institution, described the Oldowan toolkit as the first simple food processor, noting that the sharp-edged flakes could slice as efficiently as a lion's teeth, and the hammerstones could crush as powerfully as an elephant's molars. This technological augmentation of biological capabilities likely had profound evolutionary consequences, potentially subsidizing the energy costs of larger brains and enabling populations to survive in more diverse and variable environments. Sites dating to 2.9 million years ago at Nyayanga, near Lake Victoria in Kenya, preserve direct evidence of this dietary revolution. Stone tools found alongside butchered hippopotamus remains, the massive bones bearing cut marks that match the edges of nearby flakes. Interestingly, researchers also discovered teeth from Paranthropus at Nyayanga, a robust hominin lineage that diverged from the Homo line, raising the possibility that multiple hominin species independently developed or shared toolmaking traditions. The appearance of stone tools fundamentally altered hominin ecology and potentially set the stage for the emergence of the genus Homo itself. Before stone technology, hominin ancestors relied primarily on dentition and manual dexterity to procure and process food, limiting their dietary range and competitive ability relative to specialized carnivores and herbivores. With stone tools, even species with relatively modest jaw strength and tooth size could access resources previously beyond reach. This technological breakthrough may have released evolutionary pressure on jaw and tooth morphology, allowing natural selection to favor other traits, particularly increased brain size, that consumed enormous metabolic resources but conferred advantages in problem solving, social coordination, and environmental adaptation. The earliest members of the genus Homo, appearing around 2.8 million years ago, possessed larger brains, smaller jaws, and more modern body proportions than their Australopithecus predecessors, changes that align temporally with the proliferation of Oldowan tool sites across East Africa. Whether stone tool use drove these anatomical changes or simply accompanied them remains debated, but the correlation is undeniable. What began with crude hammerstone strikes on cobbles beside a Kenyan river 3.3 million years ago ultimately catalyzed a cascade of biological and cultural evolution that would, millions of years later, lead to spacecraft, symphonies, and civilizations. The dawn of toolmaking was, in a very real sense, the dawn of humanity's technological future. The physical anatomy required for effective stone tool manufacture reveals fascinating insights into hominin evolution. Homo habilis, living between 2.3 and 1.4 million years ago, possessed hand proportions distinctly different from earlier Australopithecines and more similar to modern humans. Analysis of the OH7 hand bones, discovered at Olduvai Gorge in 1960, found at the same stratigraphic level as stone tools, showed that this species had developed what anatomists call a precision grip, the ability to oppose the thumb and fingers in a controlled, forceful manner. This seemingly simple capability required a complex suite of anatomical features: broad fingertips to distribute force evenly, a long thumb relative to finger length to enable adequate opposition, and specific muscle architecture, including a well-developed opponens pollicis muscle that cups the radial side of the hand. Comparative studies involving modern humans replicating Oldowan tool production have identified several specialized grips essential for effective napping, including the three-jaw chuck grip that stabilizes the core stone using thumb and finger pads while keeping working edges exposed, and the cradle grip that cups irregularly shaped stones securely. These forceful precision grips require simultaneous recruitment of multiple intrinsic hand muscles and allow toolmakers to withstand the substantial percussive forces generated when striking one stone against another without crushing fingers or losing control of the core. The flexor pollicis longus muscle, which flexes the thumb's distal joint, proves particularly important for maintaining firm grips during repetitive tool use, though this muscle fatigues relatively quickly under sustained high-force contractions. Homo habilis exhibited a brain size averaging 600 to 700 cubic centimeters, substantially larger than the approximately 450 cubic centimeters typical of Australopithecus, but still considerably smaller than the 1,350 cubic centimeters average for modern humans. This intermediate brain size corresponded with intermediate cognitive capabilities sufficient for producing oldowan tools through direct percussion, but apparently not advanced enough for the more sophisticated aculine technology that would emerge later with Homo erectus. Oldowan tool production does not require extensive advanced planning or mental templates of final tool forms, as evidenced by the lack of standardization in tool shapes across different sites. Knappers selected appropriate stones and struck them to produce sharp flakes, but the specific resulting shapes varied considerably depending on the raw materials available and the particular fracture patterns of each strike. At Olduvai Gorge, spherical stones called spheroids appear frequently, likely because the local abundance of large, soft quartz and quartzite pieces lent themselves to this form, whereas at Kubifora in Kenya, where hard basalt predominates, spheroids are rare. This opportunistic approach contrasts sharply with later Acheulean hand axes, which display remarkable consistency in form across vast distances and time spans, suggesting that their makers conceived of standardized designs before beginning manufacture. Most oldowan tools show little evidence of retouching, the careful secondary flaking used to refine edges, implying that early toolmakers often used implements immediately after creation and then discarded them rather than curating and maintaining tools for repeated use. The ecological and dietary significance of these tools cannot be overstated. Chemical analysis of Homo habilis fossils indicates that the species consumed a predominantly vegetarian diet, but incorporated meat as an important supplement. The Oldowan toolkit facilitated access to animal resources, primarily through scavenging rather than active hunting. Early Homo likely encountered carcasses killed by large carnivores and used stone tools to quickly butcher meat and extract marrow before competing scavengers arrived. Cut marks on fossilized animal bones from sites containing Oldowan tools confirm that these implements were used to deflesh carcasses, dismember limbs, and strip meat from bones. The ability to crack open long bones to access calorie-dense marrow represented a particularly significant advantage, as this resource remained available even in partially scavenged carcasses that predators had largely consumed. The environmental context for these behaviors involved increasingly open grassland habitats, as East Africa's climate became progressively cooler and drier during the Pleistocene epoch. In these more arid landscapes, traditional plant foods may have become patchier and less reliable, creating selective pressure for dietary flexibility and the adoption of new feeding strategies, including tool-assisted scavenging. The cognitive demands of toolmaking, combined with the dietary benefits tools provided, likely created a positive feedback loop. Better nutrition supported larger brains, which in turn enabled more sophisticated tool use and broader dietary exploitation, which further improved nutrition in an escalating cycle that would eventually produce the genus Homo's later, more encephalized species. By 1.4 million years ago, Homo habilis had disappeared from the fossil record, likely outcompeted or absorbed by more advanced hominin species, including Homo erectus and Homo ergaster, which possessed significantly larger brains, more efficient bipedal locomotion, and the capacity for more sophisticated Acheulean tool technology. Yet the legacy of these early handy humans endured in the technological and cognitive foundations they established. The Oldowan tradition, though simple by later standards, represented humanity's first sustained technological culture, a set of learned behaviors transmitted across generations and maintained with remarkable consistency for hundreds of thousands of years. Every subsequent technological advancement, from Acheulean hand axes to Levallois-cores to microblades, to polished stone to metallurgy to microprocessors, traces its conceptual ancestry back to those first deliberate strikes on Kenyan lake shores millions of years ago. The dawn of toolmaking was not merely the beginning of technology, It was the moment when hominins began systematically externalizing cognitive processes into physical objects, creating tools that extended biological capabilities and establishing the fundamental human practice of solving problems through material innovation. The genus Homo emerged approximately 2.8 million years ago in East Africa, marking a profound transition in hominin evolution that would eventually lead to modern humanity. This evolutionary threshold didn't arrive as a sudden transformation, but rather as a gradual accumulation of anatomical, neurological, and behavioral changes that distinguished early Homo from their Australopithecus ancestors. The earliest confirmed Homo fossils come from the Ledi Jararu research area in Ethiopia, where a partial mandible, lower jaw, designated LD351, was discovered in sediments dating to 2.8 million years ago. This jaw displayed a blend of primitive and derived features. The overall shape remained relatively primitive, recalling Australopithecus morphology, yet the teeth showed reduced size, and the mandibular symphysis exhibited characteristics aligning with later Homo species. The specimen's intermediate nature illustrates a pivotal moment when natural selection was actively reshaping hominin anatomy in response to changing environmental pressures and new behavioral opportunities. Around this same period, East Africa experienced significant climatic shifts characterized by progressive cooling and drying, transforming dense woodlands into more open grassland habitats. These environmental changes placed new selective pressures on hominin populations, favoring adaptations for covering longer distances between resource patches, processing tougher plant materials, and potentially exploiting new food sources, including greater amounts of animal protein. Homo habilis, the species Louis Leakey and colleagues named Handyman in 1964, lived between approximately 2.3 and 1.4 million years ago and represents one of the earliest well-documented members of the genus. Specimens assigned to this species display brain sizes ranging from 550 to 687 cubic centimeters, representing a 45% increase over typical Australopithecus cranial capacities. This expansion wasn't merely a matter of absolute size. The shape and proportions of the brain case also changed, with the frontal and parietal regions expanding more than the occipital area, suggesting reorganization of neural architecture in regions associated with planning, problem-solving, and motor control. The face of Homo habilis protruded less than in Australopithecines, and the dental arcade, the curve formed by the tooth rows, became more parabolic, approaching the form seen in later Homo species. Tooth size showed reduction compared to earlier hominins, particularly in the premolars and molars, indicating dietary shifts away from the tough, fibrous plant materials that required large, grinding teeth. The postcranial skeleton revealed a mixture of features. The arms remained relatively long compared to the legs, suggesting Homo habilis still climbed trees regularly, yet the foot bones showed adaptations for efficient terrestrial bipedalism, including a convergent big toe and longitudinal arch. This anatomical mosaic indicates a species in transition, maintaining some arboreal capabilities for safety or resource access, while increasingly committing to ground-based life. The association between Homo habilis and Oldowan stone tools has been debated extensively, particularly following the discovery at Lomekwi that pushed toolmaking back before the genus Homo even existed. Nevertheless, Homo habilis remains the first hominin for which abundant stone tools and fossils co-occur at multiple sites across significant temporal and geographic ranges. At Olduvai Gorge, tools and Homo habilis remains appear together in deposits spanning from 1.9 to 1.4 million years ago. At Kubifora in Kenya, similar associations date between 1.9 and 1.6 million years ago. The hand anatomy of Homo habilis, particularly the OH7 specimen, shows clear adaptations for forceful precision gripping essential for effective stone knapping. Beyond simply possessing the anatomical prerequisites for tool manufacture, Homo habilis lived in environments and exhibited behaviors consistent with regular tool use. Sites preserve butchered animal bones bearing cut marks from stone implements, crushed bones suggesting marrow extraction, and lithic scatters indicating repeated return to favored napping locations. Chemical analysis of Homo habilis fossils reveals a dietary signature incorporating more animal protein than Australopithecus, consistent with stone tool-assisted scavenging or hunting. The convergence of anatomical capability, archaeological, association, environmental context, and dietary evidence makes Homo habilis the most likely candidate for sustained Oldowan tool manufacture, even if the practice originated earlier with Australopithecine ancestors. Roughly contemporaneous with Homo habilis, another early Homo species existed, Homo rudolfensis, known primarily from fossils discovered east of Lake Turkana in Kenya. First described based on skull KNM-ER 1470 found in 1972, Homo rudolfensis possessed a larger brain than Homo habilis, with cranial capacities reaching 750 cubic centimeters, yet exhibited a flatter, broader face with larger teeth. Some paleoanthropologists view Homo rudolfensis as simply a large variant of Homo habilis, while others consider it a distinct species representing a separate evolutionary lineage within early Homo. The taxonomic status remains contentious partly because the fossil record for both species is fragmentary, making definitive comparisons difficult. What seems clear is that by 2 million years ago, multiple Homo lineages coexisted in East Africa, each experimenting with slightly different combinations of anatomical features and possibly occupying subtly different ecological niches. This diversity mirrors patterns seen earlier with multiple Australopithecine species cohabiting the same regions, suggesting that hominin evolution proceeded not as a linear progression, but as a branching, experimenting process with multiple contemporaneous populations. Some lineages persisted and gave rise to descendant species. Others reached evolutionary dead ends and vanished without descendants. The evolutionary bush that was early Homo contained far more branches than the single trunk implied by simplified popular representations of human evolution. Around 1.9 million years ago, a new hominin species emerged that would dramatically expand the geographic range of the genus Homo beyond its African cradle. This species, known as Homo erectus in Asia and often designated Homo ergaster in Africa, by researchers who recognized them as separate taxa, represented a major evolutionary leap in anatomy, behavior, and adaptive capability. The distinction between Homo ergaster and Homo erectus remains taxonomically contentious. Some paleoanthropologists view African specimens as sufficiently different from Asian fossils to warrant separate species status, while others consider all these populations as geographic variants of a single widespread species. For clarity, African early forms will be discussed primarily as Homo ergaster, while later Asian populations will be referenced as Homo erectus, though acknowledging the debate continues. The most spectacular fossil evidence for this species comes from specimen KNM-WT 15,000, discovered in 1984 by fossil hunter Kamoya Kimo along the Nariokotome River near Lake Turkana in Kenya. This skeleton, known as Turkana boy or Nariokotome boy, dates to approximately 1.5 to 1.6 million years ago and represents the most complete early Homo skeleton ever recovered, preserving nearly the entire cranium, vertebral column, ribs, pelvis, and limb bones. The individual died between ages 7 and 11. Estimates vary depending on whether dental maturity or skeletal development is prioritized, and whether modern human or chimpanzee growth patterns provide the better comparison model. Despite dying young, The skeleton revealed profound insights into the species' anatomy and development. Turkana boy stood approximately 160 centimeters tall at death, with reconstructions suggesting an adult height potentially reaching 185 centimeters if he had survived to maturity. This represented a dramatic increase from earlier hominins, Australopithecus, and Homo habilis typically stood between 120 and 145 centimeters tall. The body proportions of Turkana boy closely resembled those of modern humans adapted to hot, arid climates, with relatively long legs and narrow hips that increased surface area to volume ratios, facilitating heat dissipation in open environments. Unlike Australopithecines, whose arms remained long relative to their legs, suggesting continued climbing behavior, Turkana boy's limb proportions matched those of modern humans, indicating full commitment to terrestrial life. The pelvis was narrower than in earlier hominins, and the thorax, ribcage, showed the barrel-shaped configuration characteristic of modern humans rather than the funnel-shaped thorax of apes. These skeletal features collectively indicate that Homo ergaster had evolved into an endurance walker and potentially an endurance runner, capable of covering long distances efficiently, a critical adaptation for surviving in increasingly open, resource-sparse environments. Brain size in Turkana boy measured approximately 880 cubic centimeters, with adult Homo ergaster cranial capacities averaging 900 to 950 cubic centimeters across multiple specimens. This represented roughly 50% larger than Homo habilis and approached two-thirds the size of modern human brains. The cranial anatomy of Homo erectus ergaster displayed several distinctive features that persisted across populations and time periods. The skull was long and low, with a receding forehead and thick cranial bones. A prominent brow ridge, supraorbital, torus, projected over the eye sockets, and the occipital bone at the rear of the skull bore a pronounced ridge where powerful neck muscles attached. The face projected forward more than in modern humans, and the jaw lacked a chin, a feature that would not appear until Homo sapiens evolved hundreds of thousands of years later. The nasal bones were thinner and more everted, turned outward, than in earlier hominins, creating a more prominent nasal bridge that helped warm and humidify inhaled air and recover moisture from exhaled air. Physiological adaptations crucial for surviving in arid environments, where water conservation meant the difference between life and death. Teeth were larger than in modern humans, but smaller than in Homo habilis, continuing the trend toward dental reduction that would characterize later human evolution. The robusticity of the skull and postcranial skeleton suggests that Homo erectus led a physically demanding existence, regularly enduring mechanical stresses that would fracture the bones of modern humans. Growth and development patterns revealed through Turkana boy and other juvenile specimens indicate that Homo erectus matured more rapidly than modern humans, with shorter childhoods and faster developmental trajectories, more similar to chimpanzees, than to the extended juvenile period characteristic of Homo sapiens. This faster development implied shorter generation times and potentially briefer lifespans compared to later humans, though adults likely survived into their third or fourth decades under favorable conditions. The technological innovations associated with Homo erectus marked a profound advance over the older one tradition. Around 1.76 million years ago, a new stone tool industry appeared in the archaeological record. The Aculean, named after Saint-Acheux in France, where such tools were first systematically studied in the 19th century. The signature implement of this tradition was the hand axe, a large bifacially flaked tool shaped into a teardrop or ovate form, with a sharp perimeter edge and a more rounded base or butt for grasping. Unlike older tools, which resulted from opportunistic flaking of available cobbles, Achulan hand axes display remarkable standardization in form across vast geographic and temporal distances. Specimens from Kenya, India, and Britain, separated by thousands of kilometers and hundreds of thousands of years, exhibit strikingly similar proportions and symmetry. This consistency indicates that toolmakers conceived of an idealized mental template before beginning manufacture and systematically worked raw materials to match that template. Creating a well-made hand axe required dozens to over 100 precisely placed strikes, rotating the core to maintain bilateral symmetry and progressively thinning the implement to create a functional edge. Experimental replication studies demonstrate that manufacturing such tools demands sustained attention, hierarchical motor planning, and continuous evaluation of progress toward a goal state. Cognitive capabilities substantially beyond those required for older one production. The Acheulean toolkit also included cleavers, large tools with a straight cutting edge rather than a pointed tip, and picks, as well as continuing production of simpler flakes and cores. This technology persisted for over 1.5 million years, making it by far the longest-lasting cultural tradition in human prehistory, only gradually being replaced by more sophisticated technologies after 300,000 years ago. The geographic expansion of Homo erectus beyond Africa represents one of the most consequential dispersal events in human prehistory. By approximately 1.85 to 1.77 million years ago, these early humans had reached Dmanisi in the modern Republic of Georgia, a small medieval town situated at the confluence of two rivers, where extensive excavations have yielded extraordinary fossil evidence. The Dmanisi site has produced more than 100 postcranial fossils and 5 remarkably well-preserved skulls, referred to as Dmanisi skulls 1 through 5, making it the best-preserved collection of early Homo fossils from a single location anywhere in the world. In 2025, archaeologists announced the discovery of an additional 1.8 million-year-old jawbone at Dmanisi, further enriching the site's already extraordinary fossil record. The presence of these remains so far from Africa, appearing almost immediately after Homo erectus emerged in East Africa, indicates that dispersal occurred rapidly, and that early populations possess sufficient behavioral flexibility and technological capability to survive in entirely unfamiliar environments. The route taken by these pioneering populations remains debated. Possibilities include a northern route through the Levantine corridor, modern Israel, Lebanon, and Syria, a southern route around the Arabian Peninsula, or potentially multiple dispersals using different pathways. What seems certain is that by 1.8 million years ago, members of the genus Homo had permanently established themselves beyond Africa for the first time. The Dmanisi fossils display anatomical characteristics intermediate between African Homo ergaster and later Asian Homo erectus populations, featuring relatively small brain sizes, ranging from 546 to 730 cubic centimeters, smaller even than many. Homo habilis specimens and substantially below typical Homo erectus cranial capacities. The most complete skull, designated Dmanisi skull 5 or D4500, possesses a tiny brain of approximately 546 cubic centimeters, yet exhibits facial and dental features aligning with Homo erectus, creating considerable taxonomic controversy. Initial researchers proposed designating these fossils as a separate species, Homo georgicus, reflecting their unique combination of primitive and derived traits. Later, analyses emphasized the substantial variation among the 5 skulls, with some individuals possessing robust, heavily built crania while others showed more gracile construction. Yet all were buried in the same stratigraphic level and represent a single population. This recognition of dramatic morphological variation within a single, contemporary population has profound implications for interpreting fossil diversity elsewhere. Traits used to distinguish separate species in Africa or Asia might simply represent normal variation within geographically dispersed populations of Homo erectus. The Dmanisi hominins were associated with simple Oldowan-like stone tools rather than Acheulean hand axes, suggesting that sophisticated tool technology was not a prerequisite for dispersal out of Africa. Their presence at such high latitudes also implies behavioral adaptations for surviving seasonal environments with cold winters, though whether this involved fire use, clothing, or strategic migration between warmer and cooler zones remains uncertain. Further east, Homo erectus reached the Indonesian island of Java by at least 1.5 million years ago, as evidenced by fossils from multiple sites including Sangiran, Trinil and Ngandong. Java man, discovered by Dutch physician Eugène Dubois in 1891, represented the first Homo erectus fossils ever found and became the type specimen for the species. Dubois' excavation team uncovered a skull cap, a tooth, and a thigh bone at Trinil along the Solo River. Fossils estimated between 700,000 and 1.49 million years old. At the time of discovery, these were the oldest hominin fossils known, and Dubois controversially argued they represented the missing link between apes and humans. Initially naming them Pithecanthropus erectus, the fossils sparked intense scientific debate. Within a decade, nearly 80 publications addressed Dubois' findings, with interpretations ranging from extinct ape to primitive human to evolutionary side branch unrelated to modern humanity. Sangiran has since become Indonesia's most productive Homo erectus site, yielding remains of over 80 individuals from deposits spanning 1.5 million to approximately 110,000 years ago. Recent discoveries from the Madura Strait, made during dredging operations in 2025, revealed Homo erectus skull fragments dated to approximately 140,000 years ago, demonstrating that these populations dispersed across the Sunderland lowlands during periods of lower sea level when currently submerged areas formed terrestrial habitat. Analysis of animal bones associated with these recent finds shows cut marks on water turtle remains and broken bovid bones indicating hunting and marrow extraction, suggesting Javanese Homo erectus actively hunted rather than relying purely on scavenging. The latest Javanese Homo erectus fossils, from Ngandong, date to between 117,000 and 108,000 years ago, making Java the last known refuge of this species anywhere in the world. In China, Homo erectus populations inhabited Zhoukoudian Cave near Beijing from as early as 800,000 years ago to approximately 230,000 years ago, though the precise chronology remains contested. These populations, known as Peking man Homo erectus pekinensis, became instrumental in establishing Chinese paleoanthropology and fostered important scientific dialogue between Eastern and Western researchers. The first fossil, a tooth, was discovered in 1921, and subsequent excavations yielded skull fragments, teeth and post-cranial remains from dozens of individuals, making Zhoukoudian the most productive Homo erectus site globally. Peking man displayed classic Homo erectus morphology: long, heavily fortified skulls with prominent brow ridges, a sagittal keel running along the skull's midline, extremely thickened cranial bones, protruding faces, robust chinless jaws, and large shovel-shaped incisors. Brain volumes range from 850 to 1,225 cubic centimeters, averaging just over 1,000 cubic centimeters, falling within the lower range of modern human variation. Body size estimates suggest Peking man averaged approximately 150 centimeters tall, somewhat shorter than tropical Homo erectus populations, possibly representing adaptation to the cold northern climate. The population inhabited a predominantly steppe environment with partial forest cover, coexisting with deer, rhinoceroses, elephants, bison, buffalo, bears, wolves, and large cats. Over 100,000 stone tool pieces have been recovered from Zhoukoudian, primarily consisting of debitage, manufacturing waste, simple choppers and flakes with a few retouched implements including scrapers. The cultural capabilities of Peking man, whether they hunted or primarily scavenged, whether they manufactured clothing, whether they controlled fire, remain fiercely debated, as does the question of whether they permanently inhabited the cave or were killed and transported there by giant hyenas. By approximately 200,000 years ago, Homo erectus disappeared from China, replaced by archaic Homo sapiens populations that eventually evolved into, or were replaced by anatomically modern humans. The expansion of Homo erectus from Africa to the Caucasus, Indonesia, and China within less than 200,000 years after the species first appeared represents a dispersal velocity unprecedented in earlier hominin evolution, foreshadowing the even more dramatic global expansion that Homo sapiens would achieve hundreds of thousands of years later. The control of fire represents one of the most transformative technologies in human prehistory, fundamentally altering ecology, diet, social organization, and evolutionary trajectory. Claims for the earliest definitive evidence of fire control by members of the genus Homo range from 1.7 to 2 million years ago, though these early instances remain hotly debated. Evidence for microscopic traces of wood ash indicating controlled use of fire by Homo erectus, beginning roughly 1 million years ago, has gained wide scholarly support. Some of the earliest known traces of controlled fire were found at the Daughters of Jacob Bridge in present-day Israel, dated to approximately 790,000 years ago, where archaeologists also discovered what may be the oldest evidence for cooking: fish teeth that had been heated deep within a cave around 780,000 years ago. However, distinguishing intentionally controlled fires from naturally occurring wildfires that early hominins opportunistically exploited remains an enormous methodological challenge. Natural fires sparked by lightning, volcanic eruptions, or spontaneous combustion of organic materials were common features of Pleistocene landscapes, and hominins undoubtedly encountered and benefited from such fires long before they could reliably produce flames themselves. The critical transition occurred when populations began not just using naturally occurring fire, but maintaining it continuously, transporting embers from place to place, and eventually generating fire on demand through friction or percussion methods. This shift from opportunistic exploitation to deliberate, sustained control represents a cognitive and technological watershed that transformed the human niche. The consensus among fire researchers is that habitual, systematic fire use became firmly established between 400,000 and 300,000 years ago, when archaeological evidence for fire becomes far more widespread, frequent and convincing across multiple continents. A 2011 study published in the Proceedings of the National Academy of Sciences analyzed fire evidence from European sites and concluded that fire became a significant part of the hominin technological repertoire only after approximately 400,000 years ago. Before this threshold, fire traces appear sporadically and inconsistently across the archaeological record, occurring at some sites but not others, suggesting opportunistic use rather than reliable mastery. After 400,000 years ago, virtually every occupation site contains fire evidence: charcoal, burned bones, heat-altered stones, and structural features like hearths, indicating that fire had become indispensable to daily life. By 125,000 to 120,000 years ago, anatomically modern humans use fire nearly universally, as they do today. At Kisum Cave in central Israel, occupied between 420,000 and 200,000 years ago, researchers found evidence that hominins not only used fire routinely, but controlled temperatures precisely to heat-treat flint, improving its flaking properties for tool manufacture. Spectroscopic analysis revealed that different tool types, blades, flakes, and pot lids, were heated to specific temperatures ranging from 259 degrees to 447 degrees C, demonstrating sophisticated understanding of how thermal treatment affected stone properties. This represents technology in the fullest sense: systematic application of knowledge to solve practical problems and improve material outcomes. The advantages conferred by fire control were profound and multidimensional. Fire provided warmth, extending the geographic range humans could occupy by making cold climates survivable. It offered light, allowing activities to continue into evening hours and enabling exploitation of caves and rock shelters that would otherwise be too dark for habitation. Fire deterred predators. Large carnivores instinctively avoid flames, and maintaining fires at cave entrances or around camps protected sleeping humans from nocturnal attacks. Smoke from fires also repelled biting insects, reducing disease transmission and improving comfort. Fire enabled sophisticated hunting strategies, including controlled burns that drove game toward waiting hunters or created habitat mosaics favoring particular prey species. Perhaps most significantly, fire transformed diet through cooking. Cooking denatures proteins, breaks down tough plant cell walls, neutralizes toxins, and kills parasites and pathogens, dramatically expanding the range of consumable foods and increasing the caloric and nutritional yield from those foods. A study published in 2025 suggests that for approximately 400,000 years, humans have consistently relied on fire for cooking, a practice that fundamentally altered human biology. Cooked foods require less chewing effort and less digestive processing than raw foods, allowing evolutionary reduction in jaw size, tooth size, and gut length, changes visible in the fossil record of late Homo erectus and archaic Homo sapiens. The energy savings from cooking may have subsidized the metabolic costs of maintaining large, energetically expensive brains, creating a positive feedback loop where fire use enabled larger brains, which in turn supported more sophisticated fire management and technological innovation. The social dimensions of fire were equally transformative. A campfire creates a natural focal point around which individuals gather, facilitating face-to-face interaction during evening hours that would otherwise be spent in isolated darkness. Anthropologists studying modern foraging societies document that the richest periods for storytelling, teaching, ritual performance, and community bonding occur around fires after the day's subsistence work concludes. The hearth hypothesis proposes that regular evening fire gathering created unprecedented opportunities for cultural transmission, allowing experienced individuals to share knowledge with novices, elders to recount historical events and cultural norms, and community members to strengthen social bonds through shared narrative and ritual. Some researchers argue that this fire-centered social environment accelerated the evolution of complex language, as individuals who could communicate effectively around the hearth gained social influence and reproductive advantages. Whether language preceded sustained fire use, developed alongside it, or emerged as a consequence of fire-centered social life remains debated, but the correlation between increasingly sophisticated tool technologies, expanded geographic range, larger brains, and evidence for fire control during the Middle Pleistocene epoch suggests these developments were interconnected rather than independent. The evolution of language represents one of the most debated and difficult to reconstruct aspects of human prehistory, primarily because language leaves no direct fossil evidence. Anatomical structures associated with speech, the larynx, tongue musculature, neural pathways controlling vocalization, consist of soft tissue that decomposes rapidly after death, leaving paleontologists to infer linguistic capabilities from indirect evidence, including brain endocasts, skull base anatomy, and the hyoid bone that anchors the tongue. The FOXP2 gene, sometimes called the language gene, plays a crucial role in the neural circuitry underlying speech and language, and genomic analysis reveals that the modern human version of this gene differs from the chimpanzee version at just 2 amino acid positions. Research published in 2025 by MIT scientists suggests that human language evolved only once, arising from a single ancestral population, rather than emerging independently in multiple hominin lineages. This finding implies that all modern languages, despite their surface diversity, share fundamental structural properties inherited from this single evolutionary origin. The researchers used computational models to demonstrate that the recursive, hierarchical grammar characterizing all human languages could not have evolved multiple times independently. The probabilistic constraints make convergent evolution of such complex linguistic structure essentially impossible. If language evolved only once, then all modern humans inherited this capacity from a common ancestral population, and linguistic ability represents a defining feature that emerged relatively late in hominin evolution, rather than appearing gradually across multiple species. Estimating when language first appeared remains highly speculative, with proposals ranging from as early as 2 million years ago with Homo erectus to as recently as 50,000 to 100,000 years ago with the emergence of behavioral modernity in Homo sapiens. Evidence supporting earlier origins includes the sophisticated coordination required for successful big game hunting, the transmission of complex tool-making traditions like Acheulean hand axes across vast distances with minimal morphological variation, and the planning involved in transporting stone tools and raw materials over dozens of kilometers. These behaviors arguably require at least rudimentary linguistic communication to transmit effectively across generations. However, skeptics note that modern great apes accomplish considerable social learning and coordination without language, suggesting that gestural communication, demonstration, and imitation might suffice for early hominin cultural transmission. The hyoid bone from a 60,000-year-old Neanderthal skeleton found in Kabara Cave, Israel, is virtually identical to the modern human hyoid, suggesting that Neanderthals possessed the anatomical prerequisites for speech. Genetic evidence indicates that Neanderthals carried the same FOXP2 gene variant as modern humans, further supporting the possibility that they possessed linguistic capability. If Neanderthals, who diverged from the Homo sapiens lineage between 500,000 and 400,000 years ago, inherited language capacity from a common ancestor, then linguistic origins must predate this split. Alternatively, language could have evolved independently in both lineages, though the 2025 research suggesting single origin makes this scenario less likely. Behavioral modernity refers to a suite of traits considered characteristic of Homo sapiens, including symbolic thought, abstract reasoning, planning depth, artistic expression, personal ornamentation, ritual behavior, and technological innovation. The emergence of these traits marks the transition from archaic to modern human cognition and culture. Early models proposed a sudden creative explosion, or great leap forward, occurring around 50,000 years ago, when the European archaeological record suddenly fills with cave paintings, carved figurines, musical instruments, complex burials, and sophisticated blade technologies. This apparent revolutionary change led some researchers to propose that a genetic mutation around this time rewired the human brain, enabling symbolic thought and triggering the behavioral changes visible in the archaeological record. However, this Eurocentric model has been thoroughly challenged by discoveries from Africa and the Near East showing that many supposedly modern behaviors appeared far earlier and developed gradually, rather than explosively. Engraved ochre pieces from Blombos Cave in South Africa date to 100,000 to 75,000 years ago, representing deliberate abstract marking. Shell beads clearly used as personal ornaments appear at multiple African sites between 75,000 and 82,000 years ago. Composite tools requiring hafting, attaching stone points to wooden shafts using adhesives, date to at least 70,000 years ago in Africa. Long-distance exchange networks moving marine shells and high-quality stone hundreds of kilometers inland operated by 100,000 years ago. These findings demonstrate that behavioral modernity developed in Africa tens of thousands of years before modern humans expanded into Europe, and that the creative explosion visible in the European record reflects migration into new territories rather than sudden cognitive evolution. The relationship between anatomical modernity and behavioral modernity remains complex and contested. Anatomically modern Homo sapiens appeared around 300,000 years ago, yet unambiguous evidence for fully modern behavior appears considerably later, creating a temporal gap of 100,000 to 200,000 years. Several explanations have been proposed for this gap. Perhaps anatomical changes preceded the cultural innovations they enabled, with behavioral modernity emerging gradually as populations accumulated cultural knowledge. Perhaps behavioral modernity appeared early, but its material traces were ephemeral and rarely preserved in the archaeological record. Or perhaps behavioral modernity emerged multiple times in different populations and was repeatedly lost before finally becoming universal and irreversible. Recent research emphasizes that behavioral modernity was not a single package of traits that appeared simultaneously, but rather a mosaic of capabilities that emerged at different times and places, sometimes appearing and then vanishing before reappearing later. Fire control, language, symbolic thought, and technological sophistication developed incrementally across hundreds of thousands of years, transforming archaic Homo populations into the cognitively modern humans who would eventually spread across the entire planet and fundamentally reshape Earth's biosphere. The use of ochre, iron-rich, minerals ranging from yellow to red to brown in color, provides some of the earliest material evidence for symbolic thinking and behavioral complexity in human ancestors. Ochre appears at archaeological sites across Africa, Europe, and Asia, spanning hundreds of thousands of years, making it one of the most persistent cultural markers in prehistory. At Blombos Cave in South Africa, researchers discovered engraved ochre pieces dated to approximately 100,000 to 75,000 years ago, featuring abstract geometric patterns carefully incised into the pigment's surface. These engravings represent unambiguous intentional marking. The patterns are too regular and repetitive to result from accidental scratches, and they required sustained, deliberate effort to create. At Kafsa Cave in Israel, ochre dating to approximately 92,000 years ago was found in association with human burials and other materials, suggesting symbolic use rather than purely functional application. Geochemical analysis revealed that the ochre at Kafsa was not locally available but had been transported to the site, indicating intentional selection and curation. The ochre showed evidence of grinding and processing into powder, behaviors consistent with preparation for use as body paint, rock art pigment, or inclusion in ritual contexts. Functional explanations for ochre use include hide tanning, insect repellent, sun protection, adhesive preparation for hafting composite tools, and medicinal applications. A 2025 study examining ochre from Middle Stone Age sites demonstrated that prehistoric people used this pigment for multiple purposes simultaneously, both functional tasks like hide, processing, and symbolic applications like body decoration, making the functional versus symbolic debate a false dichotomy. The symbolic interpretation of ochre use rests on several observations beyond mere presence at sites. Red ochre appears far more frequently at archaeological sites than yellow ochre, despite both being chemically similar iron oxides, differing only in oxidation state, and both being equally available in nature. This selectivity suggests preference based on color rather than chemical properties, indicating aesthetic or symbolic criteria guided procurement. As paleoanthropologist Alison Brooks notes, there are lots of rocks that come in powdery form that aren't red and didn't get used. Ochre has importance because it signals to others. The color red carries particularly strong symbolic significance because it resembles blood, one of the most biologically important substances for any animal, making red an inherently attention-capturing, emotionally charged hue. Brooks observes that some languages possess only two basic color terms, one for red and another category for all other colors, demonstrating red's perceptual and cultural primacy across human societies. When ochre appears in burial contexts, smeared on human remains or deposited as grave goods, functional explanations become implausible. A corpse has no use for hide tanning or sun protection. The association with burials implies that survivors attributed significance to the pigment beyond its material properties, treating it as symbolically appropriate for mortuary contexts, perhaps representing blood, life force, regeneration, or spiritual power. Personal ornaments provide another critical line of evidence for symbolic cognition and behavioral modernity. In 2021, researchers described 33 perforated shell beads from Bismoon Cave in southwest Morocco, dating to approximately 142,000 to 150,000 years ago, making them the oldest known jewelry in the world. These beads were fashioned from shells of the sea snail Nassarius gibbosulus and showed clear evidence of deliberate perforation, wear patterns consistent with stringing and ochre staining. Similar Nassarius shell beads have been recovered from sites across North Africa and the Levant, including Grotta de Pigeon in Morocco dated to 82,000 years ago, Skull Cave in Israel dated to 100,000 to 135,000 years ago, and Blombos Cave in South Africa dated to 75,000 years ago. In multiple cases, the shells had been transported significant distances from their marine source environments. At Oued Jabana in Algeria, a shell bead was found at a site 190 kilometers from the nearest coastline where the species naturally occurs. This long-distance transport demonstrates that the shells held value beyond mere availability, that individuals invested effort in acquiring and curating them, and that the beads functioned as something more than expedient tools. The act of creating and wearing personal ornaments implies several sophisticated cognitive capabilities: the ability to conceive of objects as symbols representing identity, status, group affiliation, or other abstract concepts; the capacity to understand that others will perceive and interpret these symbols; and the social coordination necessary to establish shared symbolic meanings within a community. The universality of personal ornamentation in ethnographically documented human societies suggests it serves fundamental social functions related to identity signaling, coalition formation, and boundary maintenance between groups. In mobile hunter-gatherer societies, portable ornaments communicate information about the wearer's group affiliation, kinship relationships, life stage, ritual status, and individual achievements, allowing strangers to assess whether they are encountering allies or competitors. Alison Brooks emphasizes this communicative function. If you're going to walk into someone else's territory, you want to signal that you're a friend, not a foe. Anything red is extremely visual. The appearance of standardized ornament types across broad geographic regions during the Middle Stone Age indicates that populations shared cultural traditions and maintained social connections across distances, possibly through periodic aggregations, exchange networks, or exogamous marriage patterns linking distant communities. The simultaneous emergence of shell beads, ochre use, engraved objects, and sophisticated stone technology between 150,000 and 75,000 years ago across Africa and the Near East represents the archaeological signature of behavioral modernity crystallizing within Homo sapiens populations. These were people whose minds worked fundamentally like ours, capable of abstract thought, symbolic representation, future planning, aesthetic appreciation, and the complex social reasoning necessary for maintaining large-scale cooperative networks. Fire provided the material means for expanding human ecological dominance. Language supplied the communicative infrastructure for accumulating and transmitting cultural knowledge. And behavioral modernity furnished the cognitive toolkit for transforming environmental challenges into opportunities for innovation and adaptation. Neanderthals, scientifically designated Homo neanderthalensis, represent humanity's closest extinct relatives and one of the most extensively studied hominin species in paleoanthropology. Named after the Neander Valley in Germany, where the first recognized specimen was discovered in 1856, Neanderthals inhabited Europe and Western Asia from approximately 400,000 years ago until their disappearance around 40,000 to 30,000 years ago. Genetic evidence indicates that Neanderthals and Homo sapiens shared a common ancestor approximately 500,000 to 400,000 years ago, after which the lineages diverged with Neanderthals evolving in Europe and Western Asia, while early Homo sapiens developed in Africa. This relatively recent divergence means Neanderthals were far more similar to modern humans than to earlier hominin species like Homo erectus, sharing approximately 99.7% of their DNA with Homo sapiens. A genetic similarity roughly equivalent to the relationship between modern humans and chimpanzees at the species level, yet far closer in absolute terms. The anatomical and genetic proximity between Neanderthals and modern humans has fueled intense scientific debate about whether they constitute a separate species or merely a subspecies of Homo sapiens, with taxonomic classifications varying among researchers based on differing species concepts. Regardless of taxonomic designation, Neanderthals possess distinctive physical characteristics that evolved as adaptations to the cold glacial environments of Ice Age Europe Neanderthal anatomy reflected physiological adaptations for surviving in harsh, cold climates where efficient heat conservation meant the difference between life and death. Compared to anatomically modern humans, Neanderthals possess shorter limbs relative to torso length, reducing surface area and minimizing heat loss, a pattern consistent with Bergmann's rule and Allen's rule governing body proportions in cold-adapted mammals. Their bodies were exceptionally robust, with thick, heavily built bones and pronounced muscle attachment sites, indicating tremendous physical strength, exceeding that of most modern humans. Cranial capacity averaged approximately 1,400 to 1,600 cubic centimeters, actually exceeding the modern human average of 1,350 cubic centimeters. Though brain organization and internal structure differed in subtle but potentially significant ways. The Neanderthal skull exhibited distinctive morphology, including a long, low cranial vault with a pronounced occipital bun, a backward projection at the skull's rear, massive brow ridges, a receding forehead, a large protruding face and the absence of a chin. The nasal aperture was exceptionally large and the internal nasal structures extensive. Adaptations for warming and humidifying frigid glacial air before it reached the lungs. Dental analysis reveals unique features including anterior tooth wear, suggesting Neanderthals regularly used their front teeth as tools for gripping and manipulating materials, and specialized wear patterns on certain molars indicating consistent hand preference when processing food or materials. The geographic range of Neanderthals extended from the Atlantic coast of Europe eastward through the Mediterranean region, across Central Asia to the Altai Mountains of southern Siberia, and southward into the Near East, where they encountered and interbred with expanding Homo sapiens populations. Major Neanderthal fossil sites include La Chapelle-aux-Saints and La Ferracie in France, El Cedron and Zafaraya in Spain, Monte Circeo and Sacco Pastore in Italy, Shanidar in Iraq, Teshik Tash in Uzbekistan, and Denisova Cave in Russia's Altai Mountains. Population density remained low throughout Neanderthal existence, with estimates suggesting total populations across their entire range never exceeded 70,000 individuals, and may have frequently fallen below 10,000 during glacial maxima when habitable territory contracted. This chronically small population size, combined with geographic fragmentation into isolated subpopulations during cold periods, limited genetic diversity and may have contributed to eventual extinction when competing with numerically superior Homo sapiens populations migrating into Europe after 50,000 years ago. The Neanderthal mtDNA lineage shows remarkably low diversity compared to modern human populations, suggesting repeated population bottlenecks and low effective population sizes throughout their evolutionary history. The Mousterian technological tradition, named after the Le Moustier rock shelter in France, characterized Neanderthal stone tool manufacture from approximately 300,000 to 30,000 years ago. This technology represented a significant advance over earlier Acheulean traditions, featuring the Levallois technique, a sophisticated method of core preparation that allowed toolmakers to predetermine flake size and shape before detachment. In Levallois reduction, knappers carefully shaped a stone core by removing a series of preparatory flakes around its perimeter, creating a convex upper surface and flat striking platform. Once properly prepared, a single well-placed strike could detach a large, thin flake of predictable form, essentially transforming the core into a flake factory that could produce multiple standardized implements. This approach required extensive planning, understanding of three-dimensional geometry, and the cognitive ability to envision the desired end product while executing numerous intermediate steps. Capabilities indicating sophisticated abstract reasoning. Mousterian assemblages contained diverse tool types, including scrapers for processing hides, pointed flakes suitable for cutting and piercing, denticulates with serrated edges for sawing and shredding plant materials, and hand axes continuing the Acheulean tradition. Regional variation in tool type frequencies suggests different Neanderthal populations developed distinct technological traditions adapted to local environments and available resources, implying cultural transmission of learned behaviors rather than purely instinctive tool manufacture. Evidence for Neanderthal, symbolic behavior and behavioral modernity has accumulated substantially in recent decades, challenging earlier depictions of these hominins as cognitively inferior to Homo sapiens. At Grotte du Rena in France, a Neanderthal site dating to approximately 40,000 years ago, archaeologists recovered grooved bear, wolf, and fox incisors that had been deliberately modified for suspension as ornaments, along with ochre and other symbolic artifacts. Similar findings at other sites across Europe indicate that personal ornamentation was not an isolated occurrence, but part of a broader Neanderthal cultural repertoire. The debate centers on whether Neanderthals developed these symbolic behaviors independently or adopted them through cultural contact with arriving Homo sapiens populations. Even if Neanderthals acquired symbolic practices through cultural exchange, rather than independent invention, the capacity to recognize, adopt, and integrate complex cultural innovations demonstrates cognitive sophistication comparable to behavioral modernity. As researchers emphasize, if Neanderthals possessed the ability to understand and reproduce symbolic culture, then the foundations for behavioral modernity must lie deeper in the hominin lineage than previously assumed. Predating the Homo sapiens-Neanderthal split, perhaps, the most dramatic evidence for Neanderthal symbolic cognition comes from cave art dating to approximately 65,000 years ago, predating the arrival of Homo sapiens in Europe by at least 20,000 years. In 2018, researchers announced that red ochre paintings in 3 Spanish caves, La Pasiega, Maltravieso, and Ardales, were created by Neanderthals rather than modern humans. Uranium-thorium dating of calcium carbonate deposits that had formed over the paintings provided minimum ages, establishing that the art was created before anatomically modern humans reached the region. At Maltravieso Cave, red hand stencils, created by placing a hand against the wall and blowing or spattering pigment around it, date to at least 700 years ago. In La Pasiega Cave, a red ladder-shaped symbol composed of horizontal and vertical lines dates to at least 64,800 years ago. These abstract markings and handprints demonstrate that Neanderthals engaged in symbolic representation, creating images that served no obvious utilitarian function, but instead communicated meaning, marked territory, or expressed identity. The discovery fundamentally overturns the assumption that symbolic art was exclusively a Homo sapiens innovation. If both modern humans and Neanderthals independently created abstract art, then the cognitive foundations for symbolic thought must have existed in their common ancestor more than 400,000 years ago, representing a shared heritage rather than a derived trait unique to our species. Neanderthal burial practices provide another window into their cognitive and social worlds. Multiple Neanderthal sites preserve skeletons in positions suggesting deliberate burial rather than accidental burial through sediment accumulation or carnivore activity. At La Chapelle-aux-Saints in France, a Neanderthal skeleton discovered in 1908 lay in a shallow pit apparently excavated specifically for interment, with animal bones and tools found in association. At Shanidar Cave in Iraq, excavations in the 1950s and 1960s uncovered several Neanderthal burials, including one famously associated with flower pollen, leading excavator Ralph Selecki to propose that mourners had placed flowers in the grave, though subsequent researchers challenged this interpretation. The pollen might have been introduced by burrowing, rodents or wind rather than intentional human deposition. The debate itself illustrates how close Neanderthal behavior approaches the threshold of symbolic mortuary practices. Other burials show less ambiguity. At La Ferracie, multiple Neanderthal individuals, including adults and children, were buried in what appears to be a family cemetery, suggesting ongoing social memory and deliberate site reuse for mortuary purposes. The Shanidar 1 skeleton represents an adult male who survived for years after sustaining severe injuries that left him partially blind, with a withered right arm, and walking with a limp. Survival that would have been impossible without sustained care and food sharing from other group members. This individual's prolonged survival demonstrates that Neanderthal social groups supported incapacitated members rather than abandoning them, implying empathy, social bonds, and cooperative caregiving. Around 50,000 to 40,000 years ago, anatomically modern Homo sapiens migrating from Africa and the Near East began entering regions previously occupied exclusively by Neanderthals. The two populations coexisted in Europe and Western Asia for approximately 5,000 to 10,000 years, during which genetic evidence confirms they interbred. Modern non-African human populations carry approximately 1 to 4% Neanderthal DNA, indicating that interbreeding occurred multiple times during the period of overlap. Conversely, late Neanderthal genomes show traces of Homo sapiens ancestry, confirming bidirectional gene flow rather than unidirectional hybridization. This genetic exchange demonstrates that the two populations did not treat each other as entirely separate species, but rather as potential mates, at least occasionally. Whether these encounters were peaceful or involved conflict, whether they represented rare anomalies or regular interaction, and whether hybrid offspring were socially accepted or marginalized remains impossible to determine from genetic evidence alone. What is clear is that the biological boundary between Neanderthals and Homo sapiens was permeable enough to allow viable hybrid offspring who survived to reproduce, passing Neanderthal genetic legacy into the modern human gene pool where it persists today. By approximately 40,000 to 30,000 years ago, Neanderthals had disappeared from the archaeological record entirely, their extinction coinciding with the expansion of Homo sapiens populations across Europe. The causes of Neanderthal extinction remain intensely debated, with proposed factors including direct competition with numerically superior Homo sapiens populations, climate change during the last glacial maximum, creating increasingly challenging environmental conditions, disease transmission from incoming modern human populations, and assimilation through interbreeding that gradually absorb Neanderthal populations into the expanding Homo sapiens gene pool. Evidence from dietary studies suggests that during the period of coexistence, both Neanderthals and contemporary Homo sapiens populations exploited similar large game resources, potentially creating resource competition. Later, Homo sapiens populations diversified their diet to include small game, fish, birds, and a broader array of plant resources, possibly giving them ecological advantages in marginal environments where large game became scarce. The small effective population size of Neanderthals made them vulnerable to stochastic demographic events. Random fluctuations in birth rates, death rates, or sex ratios that, in small populations, can drive extinction even without external pressures. The last known Neanderthal populations survived in refugia like Gibraltar and southern Iberia until approximately 28,000 years ago, where evidence shows they employed sophisticated subsistence strategies, including marine resource exploitation, seasonal scheduling of activities, and exploitation of both large and small terrestrial fauna. These final populations demonstrated behavioral complexity matching contemporary Homo sapiens, yet they disappeared nonetheless, leaving only genetic traces in modern human DNA and a rich archaeological record documenting a species that was, in most meaningful ways, behaviorally human. Neanderthal subsistence strategies centered heavily on hunting large and medium-sized ungulates, including deer, bison, horses, wild cattle, reindeer, and occasionally mammoth. Stable isotope analysis of Neanderthal bone collagen, which preserves dietary protein signatures, demonstrates that these hominins functioned as apex predators, obtaining nearly all their dietary protein from animal sources. At Vindija Cave in Croatia, isotopic analysis of 2 Neanderthal specimens revealed nitrogen isotope ratios matching those of contemporary carnivores like wolves and hyenas, indicating that Neanderthals occupied the same trophic level and consumed similar prey. These results replicated findings from earlier Neanderthals in France and Belgium, establishing a consistent pattern across time and space of Neanderthal adaptation as high-level carnivores. The isotopic evidence makes scavenging as a primary subsistence strategy unlikely. Scavengers would exhibit more variable isotopic signatures reflecting opportunistic consumption of diverse resources, whereas the Neanderthal values indicate consistent access to fresh large-game meat. The metabolic demands of their robust, muscular physiques in cold glacial environments required enormous caloric intake. Estimates suggest adult Neanderthal males needed approximately 5,000 calories daily, comparable to modern Tour de France cyclists during competition. This extraordinary energy requirement necessitated regular access to calorie-dense foods, particularly fat and protein from large mammals. Archaeological evidence reveals that Neanderthal hunting methods were sophisticated and adaptable. Analysis of faunal assemblages from southwestern France shows that before approximately 80,000 years ago, Neanderthals primarily hunted non-migratory species like red deer and roe deer, using relatively simple dual-face stone tools and displaying limited evidence of seasonal scheduling. After 75,000 years ago, the toolkit diversified to include retouched blade flakes optimized for repeated use, and faunal assemblages showed increased exploitation of migratory species like reindeer and bison at specialized kill sites used seasonally when herds passed through particular locations. This shift indicates that later Neanderthals scheduled hunting activities around predictable seasonal movements of prey, establishing satellite hunting camps where they processed high-utility animal parts before transporting meat, grease, marrow, and hides back to main residential sites. Mortality profile analysis studying the age distribution of hunted animals reveals that Neanderthals employed both selective hunting, targeting prime age, adults, and non-selective hunting, killing animals regardless of age, suggesting tactical flexibility depending on circumstances. The presence of complete ungulate skeletons at some sites indicates Neanderthals occasionally transported entire carcasses, while other sites preserve only high-utility skeletal elements, demonstrating that they made strategic decisions about field processing versus whole carcass transport based on distance, carcass size, and available labor. Neanderthal use of fire for cooking is well documented at numerous sites, though debates continue about whether they could generate fire on demand or relied on maintaining fires captured from natural sources. Archaeological evidence from many Middle Paleolithic sites includes hearths, charcoal deposits, burned bones, and heat-altered stone tools, demonstrating that Neanderthals routinely used fire and understood its properties. Research on burned bone chemistry at sites like Kabara Cave in Israel confirms that some bones were damaged during cooking rather than post-depositional burning or refuse disposal. Neanderthals demonstrated sophisticated understanding of fuel properties, selecting different materials for specific purposes, dry-standing wood for general fires, coal for heat conduction when cooking foods or heat-treating flint, and mixtures of bone and wood for extending burn duration. This fuel selectivity indicates experimentation and accumulated knowledge about combustion properties transmitted across generations. Whether Neanderthals possessed the technological capability to create fire from scratch through friction or percussion techniques remains uncertain, with some researchers arguing that the intermittent presence of fire at sites suggests they couldn't reliably generate flames and instead maintained fires transported from natural sources or neighboring groups. Others counter that the widespread evidence for systematic fire use after 400,000 years ago, the sophisticated fuel management practices, and the colonization of cold northern latitudes all imply reliable fire-making capability rather than dependence on chance encounters with natural fires. The genetic legacy of Neanderthals persists in modern human populations, influencing phenotypes ranging from immune function to skin biology to disease susceptibility. All non-African modern humans carry approximately 1 to 4% Neanderthal DNA, inherited through interbreeding between 50,000 and 40,000 years ago when expanding Homo sapiens populations encountered Neanderthals in Europe and Western Asia. Recent studies analyzing electronic health records from tens of thousands of individuals identified specific associations between Neanderthal genetic variants and modern health outcomes. Neanderthal alleles influence risk for type 2 diabetes, Crohn's disease, lupus, cirrhosis, depression, hypercoagulation, and smoking behavior. They also affect dermatological traits including skin lesions from sun exposure, consistent with Neanderthal adaptation to the reduced UV radiation of high-latitude environments. Neanderthal DNA concentrates in genes affecting hair and skin characteristics, likely because these genes helped modern human populations adapt to non-African environments. By conferring traits evolved over hundreds of thousands of years of Neanderthal residence in these regions. Conversely, Neanderthal DNA is conspicuously depleted in regions of the X chromosome and in genes expressed specifically in testes, suggesting that natural selection eliminated Neanderthal variants that reduced male fertility or caused incompatibilities between Neanderthal and modern human genomes. This pattern indicates that while interbreeding occurred and some Neanderthal alleles proved adaptive, genetic incompatibilities existed that natural selection purged over subsequent generations. The extinction of Neanderthals between 40,000 and 30,000 years ago closed a lineage that had endured for approximately 400,000 years across diverse and often harsh glacial environments. Their disappearance cannot be attributed to a single cause, but likely resulted from multiple interacting factors, including climate change during the last glacial maximum, competition with numerically superior Homo sapiens populations for increasingly scarce resources, possible disease transmission, low genetic diversity due to small population sizes, and gradual genetic absorption through interbreeding. The archaeological record shows no evidence that Neanderthals were cognitively or behaviorally inferior to contemporary Homo sapiens. They created abstract art, practiced symbolic behavior, manufactured sophisticated tools, controlled fire, hunted effectively, cared for disabled group members, and buried their dead. Yet despite these capabilities, they vanished while Homo sapiens populations flourished and expanded. The Neanderthal chapter in human evolution ultimately underscores both the cognitive sophistication that evolved deep within the hominin lineage before the modern human species appeared, and the precariousness of hominin populations facing environmental change and competition from closely related species, occupying overlapping ecological niches. The appearance of Homo sapiens marks the most recent chapter in a multi-million-year evolutionary story that began with the first tool-making hominins beside African lakes. Anatomically modern humans, possessing the skeletal proportions, cranial morphology, and brain organization characteristic of living people, emerged in Africa approximately 300,000 years ago. Though pinpointing a precise moment of speciation proves conceptually problematic, given that evolution operates through gradual population-level changes rather than sudden transformations. The earliest fossils assigned to Homo sapiens come from Jebel Irhoud in Morocco, dated to roughly 315,000 to 280,000 years ago, and include partial crania, mandibles, teeth, and limb bones from at least 5 individuals. These specimens display a mosaic of archaic and modern traits. The face appears essentially modern in its flat, retracted configuration, yet the brain case remains elongated and less globular than in contemporary humans, indicating that cranial evolution toward fully modern morphology continued for tens of thousands of years after the Jebel Irhoud population lived. Other early Homo sapiens fossils include the Homo kibish, remains from Ethiopia dated to approximately 233,000 to 195,000 years ago, and the Herto skulls, also from Ethiopia, dated to around 160,000 years ago. The geographic distribution of these early fossils across North and East Africa suggests that Homo sapiens did not evolve in a single, localized population but rather emerged across a broad geographic range spanning much of the African continent. For decades, the dominant model of modern human origins posited a relatively simple narrative. Homo sapiens evolved from archaic African populations around 200,000 years ago in a circumscribed region, then dispersed outward beginning approximately 80,000 to 60,000 years ago, replacing all other hominin populations, Neanderthals in Europe, Homo erectus in Asia, and other archaic groups, with minimal interbreeding. This out-of-Africa, or recent African origin, model drew support from mitochondrial DNA studies, showing that all living humans trace their maternal lineage back to Africa within the past 200,000 years, and from anatomical studies demonstrating progressive modernization of skeletal features in African fossils, while contemporary populations elsewhere retained more archaic morphology. The competing multi-regional evolution hypothesis proposed instead that modern humans evolved simultaneously across Africa, Europe, and Asia through gene flow connecting geographically dispersed archaic populations, with regional continuity in certain anatomical traits, linking ancient and modern populations within each continent. Genetic evidence accumulated over the past 2 decades has largely refuted strong versions of multi-regionalism, while simultaneously complicating the simple out-of-Africa narrative, revealing that human evolution involved complex patterns of population structure, repeated dispersals and contractions, interbreeding between divergent lineages, and admixture between modern humans and archaic populations, including Neanderthals and Denisovans. In 2025, researchers published groundbreaking findings that fundamentally revised understanding of early Homo sapiens population history, analyzing genetic data from diverse contemporary human populations. Scientists discovered that modern humanity did not descend from a single pan-mictic ancestral population, but rather from at least 2 genetically distinct groups that had diverged from each other approximately 1.5 million years ago, long before Homo sapiens as a species even existed. These 2 ancestral lineages, which researchers termed Stem 1 and Stem 2, evolved in relative isolation from each other for over 1 million years, accumulating substantial genetic differences comparable to those separating modern humans from Neanderthals. Then, beginning roughly 300,000 to 200,000 years ago, coinciding with the earliest fossil evidence for anatomically modern features, these deeply divergent populations merged, contributing genetic material to the ancestral population that would eventually give rise to all living humans. The discovery means that every person alive today carries genetic heritage from both ancient lineages, with different modern populations retaining different proportions of Stem 1 versus Stem 2 ancestry, depending on their geographic origins and demographic history. This model explains puzzling patterns in the genetic data that previous single-origin models struggled to accommodate, including unexpectedly ancient divergence times between certain genetic lineages and anomalous distributions of genetic variants across geographic regions. The implications of this discovery extend far beyond technical genetics, fundamentally altering conceptual understanding of what modern human origins means. Rather than a clean evolutionary transition from archaic to modern morphology within a single lineage, the emergence of Homo sapiens involved the merger of lineages that had been evolving separately for over 1 million years, potentially in different regions of Africa, or possibly with one lineage having dispersed outside Africa before later returning and mixing with African populations. The researchers hypothesized that prolonged isolation allowed each stem lineage to accumulate unique adaptations. Perhaps one lineage had evolved innovations in cognitive function, social organization, or technological capability, while the other possessed different adaptive advantages, and the subsequent merger combined these complementary trait suites into a single population with enhanced fitness and behavioral flexibility. This hybrid vigor resulting from the fusion of deeply divergent lineages may help explain the seemingly sudden appearance of behavioral modernity in the archaeological record around 100,000 to 80,000 years ago, as the genetic innovations from both ancestral populations became fully integrated and expressed. The finding also means that the long-standing debate between single origin and multi-regional models was framed too simplistically. Human origins were neither a simple radiation from a single African population nor a globally distributed gradual transition, but rather a complex process involving ancient population structure, long-term separation, and eventual admixture, creating the genomic foundation for modern humanity. Brain size in anatomically modern Homo sapiens averages approximately 1,350 cubic centimeters, representing a roughly threefold increase from Australopithecus and a continuation of the encephalization trend visible throughout hominin evolution. However, recent research has revealed that brain size variation within modern human populations is substantial, with some contemporary individuals possessing brains smaller than 900 cubic centimeters, less than twice the size of large-brained chimpanzees, yet exhibiting completely normal cognitive function. This finding challenges simplistic correlations between absolute brain size and cognitive capability, suggesting that brain organization, neural connectivity, and metabolic efficiency matter as much or more than raw volume. The modern human brain is distinguished not only by size, but by shape. Compared to archaic humans, the Homo sapiens cranium is more globular and spherical, with expansion occurring particularly in the parietal and frontal regions associated with abstract reasoning, language processing, working memory, and executive function. Early Homo sapiens, like those from Jebel Irhoud 300,000 years ago, already possessed brain volumes within the modern human range, yet their brain cases remained elongated rather than globular, indicating that cranial shape continued evolving toward modern configuration for tens of thousands of years after brain size had reached contemporary levels. The globularization process, which involves expansion and reorganization of specific brain regions rather than uniform enlargement, appears to have reached its modern form only within the past 100,000 to 50,000 years. Skeletal features distinguishing anatomically modern humans from archaic predecessors extend throughout the postcranial skeleton. Modern human skeletons are notably gracile, characterized by thinner cortical bone, smoother surface features where muscles attach, and more slender overall proportions compared to the robust, heavily built skeletons of Neanderthals, Homo erectus, and archaic Homo sapiens. The modern pelvis is narrower, with gracile surface features, and the elbow and knee joints show reduced surface area compared to archaic humans. Individual fingers and toes are more slender, and limb proportions shifted toward relatively longer legs compared to torso width, creating the lankier outline characteristic of modern humans. The skull exhibits distinctive modern features including reduced brow ridges, a vertical forehead rising steeply from the face, a chin entirely absent in all other hominin species, thin cranial bones, and a retracted flat face rather than the projecting midface typical of archaic populations. Teeth are smaller than in earlier hominins, continuing the trend toward dental reduction visible since Homo erectus, with incisors and canines particularly diminished compared to archaic species. The nasal aperture is narrower than in archaic forms, reflecting differences in climate adaptation between tropical African environments where modern humans evolved, and the cold glacial regions where Neanderthals and other archaic populations adapted. The gracilization of the modern human skeleton reflects multiple selective pressures and evolutionary processes. Slender limb proportions enhance thermoregulation in hot climates by increasing surface area to volume ratios, facilitating heat dissipation, a critical adaptation for endurance running and persistence hunting strategies employed by tropical hunter-gatherers. The reduction in skeletal robusticity may also reflect decreased mechanical loading as technological innovation increasingly substituted for raw physical strength, allowing natural selection to favor energy-efficient body forms that allocated metabolic resources toward brain maintenance rather than muscle and bone mass. Some researchers propose that the suite of juvenile-appearing traits characterizing modern humans, small brow ridges, globular skulls, reduced jaws, thin bones, and retention of playful, exploratory behavior into adulthood, represents a form of self-domestication driven by selection for reduced aggression and enhanced social tolerance. According to this hypothesis, as group sizes increased and social complexity intensified, Individuals with more cooperative, less reactive temperaments gained reproductive advantages, and these behavioral changes brought associated anatomical modifications as byproducts. The juvenile facial morphology of modern humans resembles the neotenous features that distinguish domesticated animals from their wild ancestors, suggesting that similar developmental processes may have operated during recent human evolution. The long temporal gap between the appearance of anatomically modern skeletal features around 300,000 years ago and the emergence of unambiguous behavioral modernity around 100,000 to 80,000 years ago has puzzled paleoanthropologists for decades. Early Homo sapiens possessed brains within the modern size range, yet the archaeological record shows they manufactured Middle Stone Age tools technologically indistinguishable from those made by archaic African populations, without clear evidence for symbolic artifacts, personal ornaments, long-distance exchange networks, or other markers of behavioral modernity. Some researchers interpret this gap as indicating that anatomical modernity was necessary but not sufficient for behavioral modernity. The neural hardware had evolved, but the cultural software required additional tens of thousands of years to develop through accumulated innovations and social learning. Others argue that behavioral modernity appeared much earlier but left minimal archaeological traces until population densities increased and environmental conditions favored preservation of organic materials and symbolic artifacts. The 2025 discovery that modern human ancestry involves the merger of two deeply divergent lineages provides a potential genetic explanation, perhaps the full suite of cognitive capabilities characterizing behavioral modernity require genetic contributions from both ancestral populations, with the integration process taking tens of thousands of years as admixture progressed and favorable gene combinations became fixed through selection. Regardless of mechanism, by 100,000 years ago, anatomically and behaviorally, modern humans had emerged in Africa, possessing the cognitive toolkit, technological capacity, and social organization that would enable their descendants to populate every habitable continent and transform Earth's ecosystems more profoundly than any species in the planet's history. The Middle Stone Age of Africa, spanning approximately 300,000 to 30,000 years ago, witnessed technological and behavioral innovations that set early Homo sapiens apart from their contemporaries elsewhere. This archaeological period represents the developmental context within which anatomically modern humans emerged, evolved behavioral modernity, and eventually dispersed beyond Africa to populate the world. Middle Stone Age stone tool technology shows a distinctive pattern of innovation appearing, disappearing, and reappearing across different sites and time periods, rather than following a linear progressive trajectory toward increasing complexity. This episodic pattern suggests that technological innovations were repeatedly invented, then lost as populations declined, moved or environmental conditions changed, then reinvented later by different populations, a process implying that environmental and demographic factors, rather than cognitive limitations, drove technological variability. Beginning around 300,000 years ago, the largest Hulian hand axes that had dominated African toolkits for over 1,000,000 years were gradually replaced by Levallois prepared core technologies that allowed production of standardized flakes and points. Blades, long, parallel-sided flakes, at least twice as long as they are wide, appear intermittently throughout the Middle Stone Age, with early examples from Kaptharin Formation in Kenya dated to 550,000 to 500,000 years ago, and at Kathupan in South Africa, demonstrating that blade technology was not a late innovation, but appeared early and persisted throughout the period. By approximately 164,000 years ago at Pinnacle Point in South Africa, early humans were systematically heat-treating silkrete stone to improve its flaking properties, deliberately heating cobbles to specific temperatures that altered the stone's internal structure and made it easier to nap into sharp-edged tools. This pyrotechnology demonstrates understanding of how thermal energy transforms material properties. Knowledge that required experimentation, accumulated experience transmitted across generations, and conceptual reasoning about cause and effect relationships not immediately visible. At approximately 70,000 years ago, the production of microlithic tools, tiny stone implements designed as replaceable components for composite-hafted weapons, became common at sites like Pinnacle Point, Sibidu Cave, and Deep Kloof Rock Shelter. These microliths were attached to wooden or bone shafts using sophisticated adhesives made from plant resins, often mixed with ochre, creating composite tools that combine materials with complementary properties. Flexible organic shafts that could be reused repeatedly, and sharp, replaceable stone edges that could be swapped out when damaged. Manufacturing these adhesives required processing tree resins through heating, sometimes using controlled pyrolysis to extract compounds, then mixing them with ochre or other materials in precise proportions, a multi-step chemical process demonstrating advanced understanding of material transformation. Blombos Cave on South Africa's southern coast has yielded some of the most spectacular evidence for early symbolic behavior and technological sophistication. Occupation layers dated between 100,000 and 70,000 years ago preserve engraved ochre pieces bearing abstract geometric patterns, parallel lines, cross-hatching, and other designs that clearly resulted from deliberate carving rather than random scratching. One piece, dated to approximately 77,000 years ago, displays 9 fine lines deliberately engraved in a cross-hatched pattern, representing what some researchers argue is the oldest known abstract drawing. In 2018, researchers announced the discovery of an even older cross-hatched drawing created approximately 73,000 years ago on a silkrete flake using an ochre crayon, extending evidence for symbolic marking further back in time. At the 100,000-year-old level, archaeologists discovered 2 complete ochre processing toolkits preserved in situ, apparently abandoned shortly after use, and then buried by wind-blown sand that sealed them in place. Each toolkit consisted of abalone shells, Haliotis medae, containing residues of a pigment-rich compound, along with grinding stones, hammerstones, bone fragments, charcoal, and ochre pieces. Chemical analysis revealed that the ancient inhabitants had created a liquefied ochre mixture, possibly heated to extract fats from bone marrow, mixed with charcoal and other ingredients following what appears to be a deliberate recipe, then stored the compound in shells for later use. The complexity of this process, procuring raw materials from diverse sources, following a multi-step manufacturing sequence, possibly using fire to facilitate chemical transformations, and storing the final product in containers for future application, demonstrates planning depth, chemical knowledge, and conceptual sophistication that researchers describe as a benchmark in the cognitive evolution of Homo sapiens. The geographic distribution of Middle Stone Age sites reveals that early Homo sapiens populations successfully occupied diverse and often challenging environments across Africa. Sites are found along coastal regions where inhabitants exploited marine resources including shellfish, seals, and fish. In the interior savannas and grasslands where large mammal hunting predominated. In mountainous areas, and even at high-altitude locations like Fincha Habera in Ethiopia, where populations adapted to glaciated alpine environments over 3,000 meters above sea level. This environmental breadth contrasts with the more restricted geographic ranges of earlier hominin species and demonstrates the adaptability that would eventually enable Homo sapiens to colonize environments as diverse as Arctic tundra, tropical rainforests, high-altitude plateaus and arid deserts. Recent discoveries from the Kalahari Basin in southern Africa's interior have revealed Middle Stone Age sites far from coastal regions, challenging models that emphasized coastal environments as primary centers of human behavioral evolution. The rock shelter at Gamahana Hill in the Kalahari preserved stratified archaeological deposits, documenting occupation by early Homo sapiens deep in Africa's interior, demonstrating that the emergence of modern human behavior involved populations across the continent rather than being concentrated in a single region. By 78,000 to 67,000 years ago at Pangaya Saidi in Kenya, characteristically modern behaviors including manufacture of shell beads, bone tools, arrows, and use of ochre pigment had become well-established features of human culture. These innovations, appearing independently across multiple regions of Africa during the late Middle Stone Age, document the crystallization of behavioral modernity within the species that would soon begin its expansion beyond Africa to eventually inhabit every continent except Antarctica between approximately 80,000 and 60,000 years ago. Anatomically and behaviorally modern, Homo sapiens began dispersing from Africa in movements that would ultimately lead to the colonization of every habitable continent on Earth. This out-of-Africa migration represents one of the most consequential events in prehistory, transforming a regional African species into a global phenomenon that would eventually dominate Earth's terrestrial ecosystems. The timing, routes, and drivers of this dispersal have been reconstructed through converging lines of evidence from genetics, archaeology, paleoclimate studies, and fossil discoveries, painting a picture far more complex than early models suggested. Rather than a single exodus, the evidence now indicates multiple dispersal events, some successful and others ending in extinction, with populations repeatedly expanding during favorable climatic windows and contracting during periods of environmental stress. The genetic legacy of these ancient movements persists in contemporary human populations. Patterns of mitochondrial DNA, bichromosome lineages, and autosomal genetic variation all preserve signatures of ancient population splits, migrations, bottlenecks, and admixture events that occurred tens of thousands of years. The primary route for human dispersal out of Africa, likely followed a northern path through the Levantine corridor, the land bridge connecting northeastern Africa to southwestern Asia through present-day Egypt, Israel, Palestine, Lebanon, and Syria. This route had been used previously by Homo erectus nearly 2 million years earlier, and by later archaic human populations that moved between Africa and the Near East during warmer interglacial periods when the Sahara became less formidable. Archaeological and fossil evidence confirms that anatomically modern humans occupied sites in the Levant by at least 120,000 years ago, with specimens from Skul and Kafze caves in Israel representing early Homo sapiens populations living alongside or in close proximity to Neanderthal groups. However, these early forays into the Near East appear to have been dead ends. The populations either died out or retreated back into Africa, leaving no genetic contribution to later humans who successfully colonized Eurasia. The successful out-of-Africa dispersal that gave rise to all non-African modern human populations occurred considerably later, sometime between 80,000 and 60,000 years ago, when one or more African populations expanded beyond the continent and established themselves permanently in Asia. A southern coastal route has also been proposed, following the shores of the Red Sea and Arabian Peninsula, then tracking along the coastlines of South Asia towards Southeast Asia and Australia. Proponents of this model note that during periods of lowered sea level associated with glacial periods, much of the continental shelf was exposed as dry land, creating an expanded coastal plain that would have provided rich marine and estuarine resources for migrating populations. Modern sea level rise has submerged these ancient coastlines, taking much of the archaeological evidence with them and making the coastal migration hypothesis difficult to test directly. However, the genetic evidence shows that some of the earliest branches of the human family tree outside Africa appear in South and Southeast Asian populations, suggesting rapid movement along southern routes before populations expanded northward into other regions. Archaeological sites in Arabia dating to the appropriate time frame are rare, but discoveries of Middle Paleolithic tools at several locations across the Arabian Peninsula indicate that humans were present in this region during windows when climate was more favorable and the landscape supported vegetation and water sources. The question of what triggered the dispersal after tens of thousands of years of African residence has generated considerable debate. Some researchers emphasize cognitive and behavioral changes, perhaps the full suite of behavioral modernity, including sophisticated language, abstract thinking, advanced planning, and technological innovation, reached a threshold that enabled humans to adapt to new environments more effectively than earlier populations. The recent discovery that modern human ancestry involves a merger of two deeply divergent populations provides a potential genetic explanation, perhaps the cognitive and behavioral capabilities, enabling global dispersal only fully manifested after the genetic contributions from both ancestral lineages became integrated. Environmental factors likely played crucial roles as well. Periods of climate change created new opportunities and pressures. During humid phases, the Sahara and Arabian deserts became more passable with expanded vegetation and water sources, creating corridors for movement. During arid phases, population densities in favorable regions increased, potentially triggering dispersal as groups sought new territories. A 2013 study demonstrated that major pulses of technological innovation in Middle Stone Age, South Africa coincided with rapid shifts toward more humid conditions in southern Africa, simultaneous with droughts in northern sub-Saharan regions, suggesting that climate-driven environmental changes created both push factors, forcing populations to move, and pull factors, making new regions attractive. Genetic studies have revealed that all non-African human populations descend from a relatively small founding group that left Africa, creating a genetic bottleneck visible in reduced genetic diversity outside Africa compared to within the continent. African populations harbor approximately twice the genetic diversity of any non-African population, consistent with a longer evolutionary history in Africa and a founder effect as small migrating groups carried only a subset of Africa's total genetic variation into new territories. Furthermore, genetic distances between populations increase with geographic distance from Africa, a pattern consistent with serial founder effects as populations dispersed outward in stages, with each new colonization event involving a subset of the preceding population and therefore carrying progressively less genetic diversity. The mitochondrial DNA of all living non-Africans coalesces to lineages that trace back to Africa between 60,000 and 80,000 years ago, providing a molecular clock estimate for the timing of dispersal that aligns remarkably well with archaeological and fossil evidence. Y-chromosome phylogenies tell a similar story, with non-African Y-chromosome lineages forming a subclade nested within the broader diversity of African Y-chromosome variation. Once modern humans had established themselves in Southwest Asia, dispersal proceeded rapidly along multiple routes. By approximately 65,000 to 50,000 years ago, the timing remains debated, humans had reached Sahel, the combined landmass of Australia, New Guinea, and Tasmania that formed a single continent during periods of lowered sea level. This colonization of Australia represents one of the most remarkable achievements of early human dispersal, because it required deliberate open-water sea crossings even when sea levels were at their lowest. The shortest water gap between the Southeast Asian islands and Sahul still exceeded 90 kilometers, meaning that the founding populations must have possessed watercraft capable of carrying people and supplies across substantial stretches of open ocean, making them among the world's earliest known mariners. Archaeological evidence from the Majid Bebe rock shelter in northern Australia, published in 2017, pushed back the date of human arrival to at least 65,000 years ago, based on luminescence and radiocarbon dating of artifacts, including stone tools, grinding stones, ochres, and intact fireplaces. However, recent genetic studies analysing Neanderthal DNA in modern Aboriginal Australian populations suggest the arrival may have occurred closer to 50,000 years ago, after the pulse of Neanderthal modern human interbreeding that occurred in Europe between 50,500 and 43,500 years ago. The debate continues, with some researchers arguing that archaeological evidence firmly supports the earlier date while others emphasize constraints imposed by genetic data. The colonization of East Asia followed complex pathways, with populations reaching southern China possibly as early as 80,000 to 70,000 years ago and expanding into northern regions by approximately 45,000 years ago. The Xiyu site in northern China, dated to around 45,000 years ago, preserves a remarkable technological assemblage that demonstrates the complexity of human cultural adaptation during dispersal. Stone tools at Xiu show a blend of technologies. Levallois prepared core methods characteristic of Middle Paleolithic traditions, blade reduction techniques typical of the Upper Paleolithic, and core flake industries found in earlier Chinese sites, suggesting cultural mixing as dispersing populations encountered resident archaic human groups or as different migrant populations with distinct technological traditions merged. Raw material studies revealed that obsidian was transported to Xiu from sources hundreds of kilometers distant, indicating sophisticated resource procurement networks and advanced planning. Unique artifacts, including a shaped graphite disc and bone tools, further demonstrate technological experimentation and innovation. This cultural blending, which researchers describe as creolization, challenges simplistic models of human dispersal as a uniform wave of technologically superior populations replacing all predecessors. Modern humans reached Europe by approximately 45,000 years ago, entering a continent that had been occupied by Neanderthals for hundreds of thousands of years. Discoveries at Ranis Cave in Germany, published in 2024, provided conclusive genetic evidence that Homo sapiens had reached northern Europe by 45,000 years ago, contemporaneous with the Linkombian-Renesian-Jerzmanovichian (LRJ) technocomplex, characterized by elongated stone points, partly shaped on both sides. DNA analysis of bone fragments from the site confirmed they belonged to Homo sapiens rather than Neanderthals, definitively identifying the makers of LRJ tools as modern humans and documenting the earliest known Homo sapiens in Central and Northwestern Europe. The presence of modern humans in cold northern latitudes by 45,000 years ago, overlapping with Neanderthals, who persisted in southwestern Europe until approximately 40,000 to 30,000 years ago, documents a period of several millennia when both species coexisted across the continent. Isotopic analysis of bones from Ranis revealed that these early European modern humans had diets dominated by terrestrial mammals, including reindeer and horse, indicating they had successfully adapted to cold steppe tundra environments. The rapid expansion of Homo sapiens populations into northern Europe shortly after their initial appearance suggests they possessed effective cold climate adaptations, including tailored clothing, sophisticated fire management, and possibly transportable shelters. The peopling of the Americas represents the final major chapter of Pleistocene human dispersal, occurring considerably later than colonization of other continents. The traditional model proposed that humans entered the Americas via the Bering Land Bridge, a broad expanse of dry land connecting Siberia to Alaska that emerged when sea levels dropped during glacial maxima around 15,000 to 13,000 years ago. These first Americans, associated with distinctive Clovis projectile points, were thought to have moved rapidly southward through an ice-free corridor between the Laurentide and Cordilleran ice sheets covering much of North America, reaching southern South America within a few thousand years. However, accumulating archaeological evidence from sites including Monteverde in Chile, dated to approximately 14,500 years ago, and possibly older sites in Brazil and Texas, indicates that humans may have arrived in the Americas earlier than the Clovis first model proposed, possibly traveling along Pacific coastal routes before interior pathways opened. Genetic studies of Native American populations reveal that most indigenous peoples of the Americas descend from a single, founding population that diverged from Asian populations and then experienced a period of genetic isolation, possibly in Beringia itself during the last glacial maximum, before expanding into the American continents as ice sheets retreated. The successful colonization of every habitable landmass except Antarctica within roughly 60,000 years of leaving Africa represents an extraordinary testament to the behavioral flexibility, technological innovation, and adaptive capacity that characterized anatomically modern Homo sapiens. The dispersal of modern humans from Africa involved not just geographic expansion but genetic encounters with archaic human populations already inhabiting Eurasia. As Homo sapiens populations moved into Europe and Western Asia, they encountered Neanderthals. As they expanded into Eastern Asia and Oceania, they met Denisovans, a mysterious hominin group known almost entirely from genetic evidence rather than abundant fossils. Denisovans are named after Denisova Cave in Siberia's Altai Mountains, where in 2010 scientists sequenced DNA from a finger bone and discovered it belonged to a previously unknown hominin lineage that had diverged from Neanderthals and modern humans hundreds of thousands of years earlier. Unlike Neanderthals, whose fossil record includes hundreds of specimens from dozens of sites, the Denisovan fossil record consists only of that finger bone, a jawbone found on the Tibetan Plateau, some teeth, and skull fragments. Yet their genetic legacy persists in modern populations across a vast geographic range from Siberia to Southeast Asia to the Pacific Islands. Genetic analysis reveals that modern humans interbred with Denisovans in at least 3 separate encounters involving distinct Denisovan populations, each leaving different genetic signatures in present-day populations. Melanesians, Aboriginal Australians, and Filipinos carry approximately 5% Denisovan DNA, the highest proportion found in any living population. Populations in South Asia, including people in India, Nepal, Bhutan, and Tibet, also carry significant Denisovan ancestry, though at lower frequencies. The Denisovan genetic contribution to modern humans includes genes that conferred adaptive advantages in specific environments. The EPAS1 gene, found in Tibetan populations and derived from Denisovans, enhances oxygen efficiency at high altitudes by regulating hemoglobin production, allowing Tibetans to thrive at elevations where other populations suffer altitude sickness. The TBX15 and WARS2 genes, also of Denisovan origin, help Inuit populations metabolize fats more efficiently and generate body heat more effectively, adaptations crucial for surviving Arctic environments. In 2025, researchers identified another Denisovan-derived gene, MUC19, that appears in high frequencies among indigenous American populations and seems to have provided survival or reproductive advantages during the peopling of the Americas. Statistical analysis showed that MUC19 occurs in unusually high frequencies in ancient indigenous American populations, appearing in DNA from 23 individuals recovered from archaeological sites spanning Alaska to Mexico, and persists in very long stretches of archaic DNA, both indicators that natural selection favored this variant. The function of MUC19 remains incompletely understood, but its consistent presence across American populations suggests it provided significant adaptive benefits. These findings demonstrate that interbreeding with archaic human populations wasn't merely a historical curiosity but a source of genetic variation that helped modern humans adapt to novel environments as they dispersed across the globe. The out-of-Africa dispersal created a genetic bottleneck that permanently reduced genetic diversity in non-African populations. A bottleneck occurs when a population's size contracts dramatically, causing rare genetic variants to be lost while common variants persist, resulting in descendant populations with substantially less genetic diversity than the ancestral population. Evidence from multiple genetic systems, mitochondrial DNA, Y chromosomes, autosomal DNA, and microsatellite markers, all point to a severe bottleneck associated with the migration out of Africa approximately 60,000 to 50,000 years ago. Researchers using sophisticated statistical modeling identified 2 distinct bottleneck events. The first occurred as populations migrated from Africa to the Middle East, and the second occurred approximately 19,000 kilometers away, when populations crossed the Bering Land Bridge into the Americas. These bottlenecks were likely caused by geographic obstacles, seas, mountain ranges, glaciers, deserts, that held back large portions of migrating populations, while smaller groups pushed forward into new territories. Climate fluctuations that periodically opened and closed migration routes would have amplified these effects, creating temporal barriers that allowed only small numbers to pass through during favorable windows. The result is that modern non-African populations possess approximately half the genetic diversity of African populations, a reduction visible in every genetic system examined. This pattern of declining genetic diversity with increasing geographic distance from Africa, known as isolation by distance, reflects serial founder effects as successive migrating groups carried progressively smaller subsets of humanity's total genetic variation, adaptive flexibility that enabled humans to colonize environments ranging from tropical rainforests to Arctic tundra to high-altitude plateaus relied on both biological and cultural mechanisms. Biological adaptations occurred through natural selection operating on genetic variation introduced through mutation, recombination, and admixture with archaic populations. Cultural adaptations, including sophisticated fire management, tailored clothing, portable shelters, food storage, and diverse subsistence technologies, allowed populations to occupy environments where biological adaptations alone would have been insufficient. The development of watercraft capable of deep ocean crossings enabled colonization of Australia and later the Pacific Islands. Projectile weapons, including spear throwers and eventually bows and arrows, increased hunting efficiency and allowed targeting of previously inaccessible prey. The invention of nets, traps, and weirs expanded subsistence strategies to include systematic exploitation of fish and small game. Evidence from sites dating between 2 million and 1.8 million years ago in East Africa shows that even very early hominins possessed remarkable environmental flexibility, repeatedly occupying areas as conditions changed, abandoning sites when they became inhospitable, and pioneering newly disturbed landscapes created by volcanic activity or other ecological disruptions. This pattern of adaptive flexibility and pioneering behavior, visible in the earliest members of the genus Homo, became amplified in anatomically modern humans who combined sophisticated technology, complex social organization, symbolic communication, and accumulated cultural knowledge transmitted across generations. By 10,000 years ago, Homo sapiens had successfully colonized every habitable landmass except Antarctica, inhabiting environments spanning nearly the entire range of terrestrial climates and ecosystems, an ecological dominance unprecedented in any other large mammal species and achieved through the unique human combination of biological evolution and cumulative cultural adaptation. The Pleistocene epoch, spanning from approximately 2.6 million to 11,700 years ago, was an age of giants. Across every continent except Antarctica, massive mammals known collectively as megafauna, defined as animals with adult body masses exceeding 44 kilograms, dominated terrestrial ecosystems in a spectacular diversity of forms that would seem fantastical if not preserved in the fossil record. These were not merely larger versions of modern species, but entirely distinct lineages that had evolved over millions of years, filling ecological niches that would later be left vacant after their extinction. The woolly mammoth, standing up to 4 meters tall at the shoulder and weighing as much as 6 tons, roamed the Mammoth Steppe, a vast grassland ecosystem stretching from Spain through Siberia to Canada that supported the highest biomass of large herbivores Earth has ever known. Saber-toothed cats, including Smilodon fatalis with canine teeth extending 28 centimeters, stalked prey across North and South America, using their iconic elongated teeth not for biting, but for precision slashing of vital arteries and windpipes after grappling prey to the ground with powerful forelimbs. The giant ground sloth Megatherium reached heights of 6 meters when standing upright and weighed up to 4 tons, making it one of the largest land mammals ever to exist. Browsing on tree leaves, its height and powerful clawed forelimbs could reach but other herbivores could not. In Australia, the Diprotodon, a marsupial resembling a gigantic wombat, weighed up to 2.8 tons and stood 2 meters tall, the largest marsupial that ever lived, grazing across the continent's interior before disappearing. Approximately 46,000 years ago, North America hosted an extraordinary assemblage of megafauna that persisted until the terminal Pleistocene extinction approximately 12,000 years ago. The Columbian mammoth, closely related to but distinct from the woolly mammoth, stood even taller, up to 4.3 meters at the shoulder, and inhabited regions from Canada to Mexico, preferring more temperate grasslands than its cold-adapted woolly cousin. Mastodons, despite superficial similarities to mammoths, represented an entirely separate evolutionary lineage with different tooth structure reflecting their browsing diet of leaves and twigs rather than the grazing habits of mammoths. The American lion, 25% larger than modern African lions, was among the largest felids ever to exist, hunting in prides across open landscapes where mega herbivores provided abundant prey. Dire wolves, heavier and more robust than modern grey wolves, hunted in packs and specialized in taking large prey, as evidenced by the thousands of individuals preserved in the La Brea tar pits in California. Carnivores that became trapped while feeding on herbivores already mired in the asphalt seeps. The giant short-faced bear, standing 3.5 meters tall on its hind legs and weighing up to 900 kilograms, was the largest terrestrial mammalian carnivore in North America, with long limbs suggesting it was capable of sustained running, unusual for bears, allowing it to pursue prey or scavenge across vast territories. Giant beavers the size of modern black bears, weighing up to 100 kilograms, constructed lodges and dams in North American waterways, engineering aquatic habitats on scales unimaginable with modern beavers. South America developed its own unique megafauna assemblage after the continent remained isolated from North America for most of the Cenozoic era, allowing distinctive lineages to evolve in isolation. When the Isthmus of Panama emerged approximately 3 million years ago, connecting the continents and enabling the Great American Biotic Interchange, species migrated in both directions, creating mixed faunas combining northern and southern elements. Glyptodonts, massive armored mammals resembling giant armadillos, reached sizes of 3 meters in length and over 2 tons in weight, encased in rigid bony carapaces that protected them from predators, but made them vulnerable to overheating and limited their mobility. Giant ground sloths like Megatherium americanum browsed on vegetation throughout the continent, using powerful clawed forelimbs to pull down branches and possibly to defend against predators, including migrating saber-toothed cats from North America. Toxodon, a bizarre herbivore roughly rhinoceros-sized but more closely related to modern horses and rhinoceroses' distant cousins, possessed massive barrel-shaped bodies and continuously growing teeth adapted for processing tough vegetation. The terror bird Phorus rhacos, though not strictly megafauna by the weight definition, stood 2.5 meters tall and represented the apex predator role in South America before mammalian carnivores. Arrived from the north flightless, birds with massive hooked beaks capable of delivering devastating strikes to prey. Eurasia's Pleistocene megafauna included both cold-adapted species of the northern mammoth steppe and temperate species. Inhabiting more southern regions, the woolly rhinoceros, covered in thick fur and possessing a flattened horn, adapted for sweeping snow aside to reach buried vegetation, ranged across the same northern grasslands as woolly mammoths, with which they shared the landscape for hundreds of thousands of years. The cave bear, substantially larger than modern brown bears, inhabited caves throughout Europe where it hibernated during winter months, leaving behind massive bone deposits that have provided paleontologists with abundant fossils documenting these impressive animals. The giant Irish elk, despite its name actually a species of deer rather than elk and not restricted to Ireland, possessed the largest antlers of any deer species ever known, spanning up to 3.65 meters from tip to tip and weighing 40 kilograms, requiring males to consume enormous amounts of calcium-rich vegetation each year to grow these impressive but metabolically expensive ornaments. The steppe bison, ancestral to both modern European bison and American bison, roamed in vast herds across the grasslands, providing staple prey for human hunters as well as for large predators including cave lions and packs of wolves. Australia's megafauna differed profoundly from those of other continents because the dominant large animals were marsupials rather than placental mammals, The continent had been isolated since separating from Antarctica approximately 30 million years ago, allowing a unique fauna to evolve independently. Diprotodon optatum, the largest marsupial ever, possessed powerful limbs, a large head, and teeth adapted for browsing on shrubs and potentially digging for roots and tubers. Procoptodon, the giant short-faced kangaroo, stood over 2 meters tall and weighed up to 230 kilograms, double the mass of the largest modern kangaroos, and possessed a single large toe on each foot rather than the characteristic split toe of modern kangaroos, suggesting different locomotion patterns possibly involving bounding at high speeds across open landscapes. The marsupial lion Thylacoleo carnifex, despite not being closely related to true cats, convergently evolved similar predatory adaptations, including retractable claws and powerful jaws equipped with blade-like teeth capable of shearing through flesh and bone. Evidence suggests Thylacoleo was an ambush predator that may have climbed trees to drop onto prey, using its powerful forelimbs and enormous thumb claws to subdue victims. Giant monitor lizards exceeding 6 meters in length, twice the size of modern Komodo dragons, hunted across the Australian landscape, representing the apex reptilian predators in an ecosystem where reptiles played roles that mammals filled on other continents. The ecological roles played by Pleistocene megafauna extended far beyond their sheer physical presence, fundamentally shaping ecosystems through processes that smaller animals could not replicate. Large herbivores functioned as ecosystem engineers, modifying vegetation structure, accelerating nutrient cycling, and creating habitat heterogeneity that supported biodiversity across multiple trophic levels. Through consumption, trampling, and physical breakage of vegetation, megaherbivores maintained open grassland and savanna habitats that would otherwise transition to closed woodland or forest. This megafaunal browsing suppressed woody plant growth, favoring grasses and herbaceous plants, and created landscape mosaics of varying vegetation structure depending on local herbivore pressure tied to water sources, migration routes, and predation risk. Meta-analysis of 297 studies examining megafauna effects across 6 continents demonstrated that wild herbivorous megafauna significantly alter soil nutrient availability, promote open vegetation structure, reduce abundance of smaller animals, and crucially, increase ecosystem spatial heterogeneity, a key driver of biodiversity across taxonomic groups and scales. The Columbian mammoth, 3 times more massive than modern elephants and over 10 times larger than domestic cattle, would have exerted ecosystem impacts proportionally greater than those of contemporary megafauna due to non-linear scaling relationships between body size and ecological influence. Nutrient cycling represents one of the most important but least visible megafaunal ecosystem services. Large animals accelerate biogeochemical cycling by consuming plant material and liberating nutrients that would otherwise remain locked in slowly decomposing leaves, stems, and woody tissue for years or decades in cold Pleistocene steppes and dry savanna environments where decomposition proceeds slowly due to low temperatures or limited moisture. Megafaunal digestive systems functioned as giant warm and moist incubating vats that accelerated nutrient release. After megafaunal extinctions, nutrients became increasingly locked in plant litter, making entire ecosystems more nutrient poor and potentially less productive. Beyond accelerating decomposition, megafauna redistributed nutrients laterally across landscapes through their feces and urine. Because of high food consumption rates, long gut residence times, and large daily movement ranges, megaherbivores transferred significant nutrient quantities between different landscape zones, eating in one location and defecating in another, effectively pumping nutrients uphill from low nutrient to high nutrient areas, and between different habitat types. Quantitative studies concluded that larger animals are disproportionately important in nutrient transfer, increasing nutrient diffusion rates by at least an order of magnitude and functioning as ecological arteries connecting different parts of the landscape. The woolly mammoth exemplified the profound cold climate adaptations that enabled megafauna to thrive in Ice Age environments. Standing up to 3.4 meters tall and weighing 6 tons, woolly mammoths were covered in dense fur consisting of a thick yellowish-brown undercoat approximately 2.5 centimeters thick for insulation, plus long outer guard hairs reaching up to 90 centimeters that shed water and snow, small ears measuring only 38 centimeters long, far smaller than the 1.5-meter ears of modern African elephants, minimized heat loss and reduced risk of frostbite. A thick layer of subcutaneous fat, up to 10 centimeters deep, provided additional insulation and energy reserves during harsh winters when food became scarce. Genomic analysis comparing high-quality woolly mammoth genomes to Asian elephant genomes identified approximately 1.4 million DNA nucleotide differences affecting over 1,600 proteins related to Arctic survival. Alterations in genes governing skin and hair development, adipose tissue storage and metabolism, and temperature perception enabled woolly mammoths to function effectively in extreme cold. The mammoth version of the TRPV3 protein, found in skin and affecting hair growth, showed reduced sensitivity to heat compared to the elephant version, consistent with adaptations promoting denser, wavier hair for superior insulation. Hemoglobin mutations improved oxygen delivery throughout the body in cold conditions and prevented freezing, similar to adaptations found in modern reindeer and musk oxen. Circadian clock genes showed alterations perhaps necessary for coping with extreme seasonal variation in daylight duration at high latitudes. The timing of cold-adapted species evolution followed the expansion of permanent polar ice and glacial interglacial climate oscillations. Research published in 2025 demonstrated that cold-adapted animals, including woolly mammoths, musk oxen, and Arctic foxes, began evolving specialized features approximately 2.6 million years ago, when permanent ice at the poles became prevalent. A critical transition occurred around 700,000 years ago when glacial periods doubled in length, creating sustained cold conditions lasting 100,000 years, rather than the shorter 41,000-year cycles that had previously dominated. This intensification of glacial conditions drove the evolution of many characteristic Ice Age species, including mammoths, with their full suite of cold adaptations. The mammoth steppe ecosystem these animals inhabited represented Earth's most extensive biome during glacial maxima, stretching from Spain through Central Asia to Canada, and supporting higher large mammal biomass than any contemporary ecosystem. This cold, dry grassland supported vast herds of woolly mammoths, woolly rhinoceroses, steppe bison, horses and reindeer, which in turn sustained populations of large predators including cave lions, wolves, and cave hyenas. The ecosystem's productivity derived from deep, nutrient-rich lower soils created by wind-deposited glacial rock flour, and from vegetation adapted to cold, dry conditions with brief but productive growing seasons. The megafaunal communities of different continents reflected biogeographic history, with each landmass developing distinctive assemblages shaped by millions of years of evolution in partial or complete isolation. Africa retained the richest megafauna diversity because it had been continuously inhabited by large mammals throughout the Cenozoic era, allowing long-term coevolution between megafauna and hominins that potentially buffered against extinction when modern humans appeared. In contrast, Australia, South America, and northern Eurasia developed unique megafaunal communities adapted to local conditions, but ultimately vulnerable when faced with the novel, selective pressure of technologically sophisticated human hunters, arriving suddenly in ecosystems where native fauna lacked previous exposure to human predation. The ecological legacy of these Ice Age giants persists in contemporary ecosystems. The wildest modern landscapes likely represent truncated, impoverished versions of Pleistocene communities with simplified food webs, altered vegetation structure, disrupted nutrient cycling, and diminished spatial heterogeneity compared to the megafauna-dominated ecosystems that characterized most of the past 50 million years of Earth history. The dietary ecology of Pleistocene Megafauna reveals sophisticated niche partitioning that allowed multiple large herbivore species to coexist in the same landscapes without excessive competition. Isotope analysis of fossil teeth, which preserves chemical signatures reflecting diet during tooth formation, demonstrates that contemporary megaherbivores consumed different plant types, occupied distinct feeding niches, and exploited varying vertical vegetation strata. In Australian Pleistocene ecosystems, calcium and strontium isotope studies revealed that giant marsupial herbivores maintained relatively small home ranges, yet exhibited distinct dietary niches within those ranges. The giant short-faced kangaroo Procoptodon showed isotope values consistent with browsing on C3 shrubs and trees, while Protemnodon, another large macropod of similar size, exhibited values suggesting greater consumption of grasses or other plant tissues with different isotopic compositions. This dietary differentiation between two large-bodied kangaroos occupying the same general habitat demonstrates how niche specialization permitted coexistence and supported the remarkable diversity of Australian megafauna before extinction. The Diprotodon, despite its massive size, showed isotopic evidence of browsing behavior rather than grazing, consuming leaves, twigs, and possibly roots rather than grass, further reducing dietary overlap with grazing specialists. Megatherium, the iconic giant ground sloth, stood approximately 6 meters tall when rearing on its hind legs, approaching the height of a modern giraffe, and weighed up to 4 tons, making it among the largest land mammals ever to exist. Despite this enormous size, Megatherium was a herbivore that browsed on tree leaves, using its powerful clawed forelimbs to pull branches within reach of its peg-like teeth adapted for grinding vegetation. The skull was low and narrow with limited brain case volume, yet the animal's massive body size and defensive capabilities, including formidable claws reaching 50 centimeters in length, provided effective protection against predators. Footprint evidence indicates Megatherium was primarily quadrupedal when walking, but capable of assuming a bipedal stance, possibly to feed on high vegetation or to intimidate rivals and predators. Charles Darwin famously collected Megatherium fossils during the voyage of the Beagle in the 1830s, and these specimens helped establish the reality of extinction. At the time, many naturalists doubted that species could disappear entirely, assuming that missing animals simply lived undiscovered in remote regions. The discovery that these enormous ground sloths were related to modern tree sloths, which weigh less than 9 kilograms, illustrated the dramatic evolutionary transformations possible over millions of years and the vulnerability of even the most physically imposing species to changing conditions. Saber-toothed cats of the genus Smilodon represent perhaps the most recognizable Pleistocene predators, with their iconic elongated canine teeth that could exceed 28 centimeters in length. 3 species are recognized: Smilodon gracilis, the smallest at approximately 55 to 100 kilograms; Smilodon fatalis, the most common North American species weighing 160 to 280 kilograms; and Smilodon populator from South America, the largest at up to 400 kilograms, comparable to modern lions. Biomechanical studies combining fossil evidence with computer modeling have revealed how these predators employed their distinctive weaponry. Contrary to earlier speculation that the elongated canines delivered crushing bites, research demonstrated that the teeth were relatively fragile and vulnerable to breakage under excessive lateral force. Instead, Smilodon used its exceptionally powerful forelimbs, more robust than those of any modern cat, to wrestle prey to the ground and immobilize it, then positioned itself to deliver precise stabbing bites to the throat or neck, using a gaping jaw that could open to 120 degrees, far wider than modern cats. The elongated canines would slice through flesh and sever major blood vessels, causing rapid, fatal blood loss. This specialized killing method was highly effective against large, slow-moving prey like bison, horses, and young mastodons, but required close ambush rather than long pursuit. Analysis of bone injuries in Smilodon fossils from the La Brea tar pits revealed that saber-toothed cats sustained more frequent injuries than contemporary dire wolves, suggesting that Smilodon faced greater risks during predation, possibly because they hunted larger, more dangerous prey, potentially hunted alone rather than in packs, and relied on high-risk close-quarters combat that brought them into direct physical struggle with prey capable of inflicting serious wounds. Whether Smilodon was social or solitary remains debated, with evidence supporting both interpretations. The massive accumulation of Smilodon fossils at the La Brea tar pits, over 2,000 individuals from a single location, led some researchers to propose pack hunting, with multiple cats becoming trapped while attempting to feed on mired herbivores. Additionally, some Smilodon skeletons show healed injuries that would have been debilitating, preventing effective hunting for weeks or months, yet the individuals survived, potentially implying that packmates shared food during recovery. However, skeptics note that the La Brea accumulations could simply reflect repeated independent trapping events over millennia, as individual cats attempted to exploit easily accessible but ultimately fatal prey. Modern solitary cats like tigers show similar accumulation patterns at sites where prey predictably appears, without requiring pack coordination. Bone histology studies examining growth rates in juvenile Smilodon revealed relatively slow development compared to modern large cats, with individuals taking longer to reach adult size, a pattern more consistent with solitary cats than social species, which typically grow faster to reach hunting competency and contribute to group activities. The debate remains unresolved, with most researchers acknowledging that Smilodon likely exhibited flexible social organization, varying between solitary and small group living, depending on local prey density, habitat structure, and population density, much as modern cougars show variable sociality in different environments. These apex predators, regardless of social structure, played crucial roles in regulating herbivore populations and shaping plant communities through top-down, trophic cascades that influenced vegetation structure across vast Pleistocene landscapes. The late Pleistocene witnessed one of the most dramatic extinction events in Earth's recent history, with approximately 65% of all megafaunal species worldwide disappearing between roughly 50,000 and 10,000 years ago. This catastrophic loss was not distributed evenly across the globe or through time, regional. Variations in timing and severity reveal a complex pattern that has fueled decades of scientific controversy about causation. In North America, approximately 72% of megafauna went extinct. In South America, the toll reached 83%, and in Australia, a staggering 88% of megafaunal species vanished. All mammals exceeding 1,000 kilograms in body mass became extinct in Australia and the Americas, and roughly 80% of such giants disappeared globally. In contrast, Africa, South Asia, and Southeast Asia experienced more moderate extinctions, retaining substantial megafaunal diversity that persists into the present. Elephants, rhinoceroses, hippopotamuses, giraffes, and large bovids continue to inhabit these regions, representing remnants of what was once a globally distributed phenomenon. The extinctions displayed an extreme size bias, disproportionately affecting large animals, while leaving small mammals, birds, reptiles, and amphibians largely unaffected. A selectivity pattern that distinguishes this event from earlier mass extinctions driven by asteroid impacts or massive volcanic eruptions, which typically affected organisms across size classes more indiscriminately. The temporal pattern of extinctions varied dramatically by region, providing crucial clues about causation. In Australia and New Guinea, megafaunal extinctions began approximately 50,000 to 46,000 years ago, closely coinciding with the arrival of the first human populations on the continent. Within several thousand years of human colonization, species including the giant wombat-like Diprotodon, the massive short-faced kangaroo Procoptodon, the marsupial lion Thylacoleo, and numerous other large marsupials had disappeared. In northern Eurasia, extinctions were staggered over tens of thousands of years between 50,000 and 10,000 years ago, with different species disappearing at different times in different regions rather than a single synchronized collapse. The woolly mammoth persisted in Siberia until approximately 10,000 years ago on the mainland, with relict populations surviving on isolated Arctic islands, most famously Wrangel Island, where dwarf mammoths endured until roughly 4,000 years ago, contemporaneous with the Egyptian pyramids and Mesopotamian city-states. In the Americas, megafaunal extinctions occurred far more rapidly, spanning only 3,000 years at most and clustering, particularly around 13,000 to 11,000 years ago, a period bracketing the Younger Dryas climatic event and the appearance of Clovis archaeological culture, with its distinctive, fluted projectile points optimized for hunting large game. This temporal coincidence between human arrival and megafaunal extinction across multiple continents represents the strongest circumstantial evidence linking humans to the disappearances. Yet the variable timing, the survival of megafauna in Africa where humans evolved, and the lack of abundant kill sites for many extinct species have kept alternative explanations viable. The ecological consequences of these extinctions extended far beyond the simple loss of impressive animals. Megafaunal extinctions triggered cascading effects throughout ecosystems, fundamentally restructuring plant communities, altering fire regimes, disrupting nutrient cycling, reducing landscape heterogeneity, and eliminating ecological functions that smaller surviving animals could not replicate. In North America, the disappearance of mammoths, mastodons, and other mega-herbivores allowed woody vegetation to expand into formerly open grasslands and parklands, transforming landscapes that had been maintained as grassy savannas through millennia of megafaunal browsing and trampling. Nutrient cycling slowed dramatically as the rapid digestive processing that mega-herbivores provided, converting plant material into nutrient-rich dung distributed across landscapes, was replaced by slower microbial decomposition of plant litter. Long-distance seed dispersal suffered as large frugivores that consumed fruits and defecated intact seeds kilometers away disappeared, leaving certain plant species with no effective dispersal mechanism. So-called evolutionary anachronisms, like avocados and honey locust, whose massive fruits evolved to be consumed by megafauna that no longer exist. Predator populations crashed as their primary prey disappeared. Saber-toothed cats, dire wolves, giant short-faced bears, and American lions all went extinct in North America shortly after the herbivores they depended upon vanished, demonstrating that megafaunal extinction propagated upward through trophic levels. The modern ecosystems we consider natural or wild are actually deeply impoverished shadows of the Pleistocene megafauna-dominated systems that characterized most of the past several million years, fundamentally altered by removals that occurred within the span of human memory and cultural transmission. The climate change hypothesis attributes megafaunal extinctions primarily to environmental transformations associated with the end of the Pleistocene epoch and the transition into the warmer, more stable Holocene. Proponents of this explanation emphasize that the terminal Pleistocene involved dramatic climatic upheavals, including rapid temperature swings, altered precipitation patterns, shifts in vegetation communities, and increased seasonality, all potentially devastating to large-bodied animals with specialized diets, slow reproductive rates, and limited behavioral flexibility. Research published in 2021 analyzed the largest assembled database of directly dated megafauna fossils and found a consistent positive correlation between megafauna population levels and global temperature proxies, with population declines corresponding to temperature decreases. This finding suggests that cooling events, rather than warming, correlated with megafaunal stress, contrary to some earlier hypotheses proposing that rapid warming caused extinctions. The Younger Dryas stadial, a cold period lasting from approximately 12,900 to 11,700 years ago, coincided with final megafauna population declines and extinctions in North America, hinting that the unique conditions of this interval, abrupt cooling, increased seasonality, rising atmospheric CO2, and major vegetation changes occurring within decades, played an important role. During the Younger Dryas, summer insolation reached one of its highest peaks of the late Pleistocene, creating extreme seasonal contrasts with short, intense growing seasons, followed by long, harsh winters. These conditions favored smaller-bodied ruminants like deer and moose, which can subsist on less diverse diets and extract nutrients efficiently during brief growing seasons, while disadvantaging larger-bodied animals with slower metabolisms, longer gestation periods, and requirements for abundant, high-quality forage year-round. Vegetation changes, driven by climate shifts, fundamentally altered the resource base upon which megaherbivores depended. As temperatures warmed at the end of the Pleistocene, the mammoth steppe, a unique cold, dry grassland ecosystem that had supported enormous herbivore biomass, fragmented and was replaced by boreal forests, wetlands, and modern tundra, none of which could sustain comparable densities of large grazers. The rapidity of these vegetation changes, occurring in some regions over less than a century, may have prevented megafauna populations from adapting or migrating to track suitable habitat. Increased atmospheric CO2 concentrations, which rose sharply during the Younger Dryas, reduced plant nitrogen content and protein quality, potentially creating nutritional stress for herbivores, even in areas where total plant biomass remained high. Some scholars argue that these climate-driven ecological changes affected not just megafauna, but entire biotic communities. 9 megafauna species survived in North America, while 37 genera went extinct, and a new bison species emerged, suggesting differential responses to changing conditions rather than wholesale collapse. Ancient DNA studies show that bison populations experienced significant losses of genetic diversity prior to human arrival in the Americas, consistent with climate-driven population bottlenecks rather than human-caused declines. Additionally, some megafauna last appearance datums, the most recently dated fossil for a given species, significantly predate human arrival in certain regions, suggesting these extinctions occurred before humans could have been responsible. The climate hypothesis also explains why Africa retained its megafauna. The continent experienced less extreme climatic upheaval than northern latitudes, and African ecosystems had not been as heavily dominated by the cold-adapted mammoth steppe community that collapsed elsewhere. However, the climate change hypothesis faces several significant challenges. Megafaunal lineages had survived multiple previous glacial-interglacial transitions over hundreds of thousands or even millions of years, experiencing comparable or more extreme climatic oscillations without extinction, raising the question of why the terminal Pleistocene transition proved uniquely catastrophic. The Eemian interglacial, approximately 130,000 to 115,000 years ago, involved warming comparable to or exceeding the Holocene transition, with sea levels rising 6 to 9 meters above present and tropical forests expanding into higher latitudes. Yet megafauna survived this warm period without major extinctions. If climate change alone drove terminal Pleistocene extinctions, the pattern should have repeated during previous interglacials, yet it did not. The size selectivity of extinctions, with massive animals disappearing while smaller species survived, is difficult to explain purely through climate change, as environmental stresses typically affect organisms across size ranges, rather than targeting the largest species selectively. Small-bodied species should theoretically be more vulnerable to rapid climate change, due to shorter generation times and smaller home ranges limiting their ability to track shifting habitats. Yet small mammals persisted while giants vanished. Regional variation in extinction timing and severity correlates more strongly with human arrival patterns than with climatic events. Australia's extinctions occurred 35,000 years before the Younger Dryas, when the northern hemisphere megafauna disappeared. Yet the human arrival hypothesis predicts this temporal offset, while the climate hypothesis struggles to explain it. These inconsistencies have led many researchers to conclude that while climate change undoubtedly stressed megafaunal populations and may have been the proximate cause for some extinctions, it cannot be the complete explanation for the global pattern of disappearances. The overkill hypothesis, first systematically articulated by Paul Martin in the 1960s, proposes that human hunting drove Pleistocene megafaunal extinctions as technologically sophisticated Homo sapiens populations colonized new continents where native fauna had no prior evolutionary exposure to human predation. According to this model, megafauna in Africa co-evolved with hominins over millions of years, allowing natural selection to favor individuals with innate wariness of bipedal predators. African elephants, rhinoceroses, and buffalo demonstrate anti-predator behaviors toward humans that may represent this long co-evolutionary history. In contrast, when modern humans first reached Australia, the Americas, and northern Eurasia, they encountered naive fauna that had never experienced human hunting and therefore lacked appropriate escape responses, making them vulnerable to overharvesting even by small human populations. Computer modeling demonstrates that even conservative assumptions about human hunting rates, each person killing only one megafauna individual per year combined, with realistic assumptions about megafaunal reproductive rates, could drive extinction within several hundred years after human arrival. The selectivity for large animals aligns with predictions from optimal foraging theory. Large-bodied prey provide higher caloric returns per hunting effort than small prey, making them preferential targets for human hunters, while smaller animals are often ignored even when abundant. Analysis comparing paleontological abundances of extinct and surviving Pleistocene megafauna with their archaeological abundances in kill sites shows that extinct taxa appear in proportion to their fossil abundance, exactly as predicted if human hunters took all available megafauna opportunistically upon encounter. This finding contradicts the associational critique, the argument that scarcity of kill sites for extinct megafauna proves humans weren't hunting them, by demonstrating that kill site frequency simply reflects fossil rarity, and that low numbers of both fossils and kill sites are consistent with human hunting driving populations to low densities before extinction. Regional analyses provide mixed support for the overkill hypothesis, with some taxa showing strong correlations between human population growth and megafaunal decline, while others do not. A study examining summed probability distributions of radiocarbon dates for well-dated North American megafauna taxa and anthropogenic materials found that 3 species, mammoth, mastodon, and horse, showed population declines correlating with increasing human populations, consistent with hunting as the extinction driver. However, 5 other taxa, camel, tapir, ground sloth, giant short-faced bear, and saber-toothed cat, showed patterns more consistent with climate-driven extinction, with population collapses occurring independently of human population trends. A 9th taxon, bison, appeared to suffer from both human hunting and climate change, with populations declining but not becoming extinct as the species adapted to changing conditions. This taxonomic variation suggests that megafaunal extinctions resulted from multiple interacting causes, rather than a single universal mechanism, with different species responding differently to the combined stresses of human predation and environmental change. The near-simultaneous extinction of predators like saber-toothed cats and dire wolves shortly after their prey disappeared suggests that at least these carnivores went extinct due to prey loss rather than direct human hunting. Humans would have targeted herbivores preferentially, and predator extinctions followed as cascading secondary effects. The debate between climate and overkill hypotheses has evolved toward recognizing that these factors likely interacted synergistically rather than operating independently. Climate change may have stressed megafaunal populations, reducing their numbers, fragmenting their ranges, and making them more vulnerable to additional mortality from human hunting. Conversely, human hunting may have prevented megafauna from adapting to changing environments by continuously reducing population sizes below thresholds necessary for maintaining genetic diversity, occupying all available habitat patches or experimenting with new food sources, computer simulations demonstrate that moderate human hunting combined with moderate climate stress can drive extinction even when neither factor alone would be sufficient, a phenomenon called synergistic extinction. Human impacts extended beyond direct killing to include habitat modification through anthropogenic fire, which early humans used to manipulate landscapes for hunting and resource management. Evidence from Australia shows dramatic increases in fire frequency coinciding with human arrival, transforming fire-sensitive vegetation into fire-adapted communities and potentially fragmenting megafaunal habitat. Cultural motivations for hunting beyond subsistence. Prestige hunting, ritual uses, competition between groups over game resources may have driven kill rates exceeding what populations could sustain even when meat was abundant. The most comprehensive analyses now suggest that the relative importance of climate versus human factors varied by species, by region, and through time, with African megafauna surviving because they co-evolved with humans and experienced less extreme climate change, while megafauna elsewhere faced the perfect storm of climatic stress and encounter with a novel, technologically sophisticated predator against which they had no evolutionary defenses. The terminal Pleistocene extinctions eliminated Earth's megafaunal diversity accumulated over millions of years of evolution, leaving a biologically impoverished world that humanity has inherited and must now decide whether to restore, preserve in its diminished state, or allow to decline further as modern anthropogenic pressures compound the ancient losses. The creation of visual art represents one of humanity's most distinctive characteristics, separating our species from all others through the capacity to render mental images into permanent physical form. Cave paintings, created by applying pigments to rock surfaces deep within subterranean chambers, preserve the earliest substantial corpus of human artistic expression, providing windows into the minds, beliefs, and symbolic worlds of Ice Age populations. These images were not casual doodles or idle decoration, but rather deliberate creations requiring significant investment of time, effort, and resources, procuring pigments, manufacturing tools, accessing difficult locations, and executing technically sophisticated imagery. The oldest known figurative cave painting, discovered in Layang Karampuang Cave on the Indonesian island of Sulawesi, dates to at least 51,200 years ago and depicts a narrative scene showing three human-like figures interacting with a wild pig, making it the oldest known example of representational art anywhere in the world. This extraordinary antiquity demonstrates that the capacity for symbolic visual expression emerged early in the history of anatomically modern Homo sapiens and spread across vast geographic distances as human populations, dispersed from Africa into Eurasia and beyond. The Indonesian painting predates European cave art by tens of thousands of years, overturning earlier assumptions that sophisticated artistic traditions first emerged in Ice Age Europe and forcing a fundamental reconsideration of when and where human symbolic culture crystallized. The Sulawesi cave art depicts therianthropes, hybrid beings combining human and animal characteristics, engaging with a Sulawesi warty pig, a species still native to the island. The three anthropomorphic figures possess human-like body proportions but animal-like features, including what appear to be beaks or snouts and possibly tails, suggesting they represent supernatural beings, shamanic transformations, or mythological narratives. The presence of narrative storytelling in the world's oldest figurative art indicates that by 51,200 years ago, humans possessed not only the technical ability to create visual imagery, but also the cognitive and cultural sophistication to conceive and communicate complex stories involving supernatural concepts, human-animal relationships, and possibly spiritual beliefs. Uranium series dating of calcium carbonate deposits that formed over the paintings provided minimum ages, establishing that the images were created at least 51,200 years ago, but could be substantially older if significant time elapsed between painting and calcite formation. The discovery challenges Eurocentric models of human cultural evolution that positioned Ice Age Europe as the cradle of artistic innovation and instead reveals that sophisticated symbolic expression emerged across multiple regions as modern humans colonized new continents. Prior to the Indonesian discoveries, the oldest known cave art came from Europe, where hundreds of decorated caves document Ice Age artistic traditions spanning from approximately 40,000 to 10,000 years ago. The Chauvet Cave in southern France, discovered in 1994 by Jean-Marie Chauvet and colleagues, contains some of Europe's oldest and most spectacular cave paintings, with radiocarbon dates indicating creation between 32,000 and 30,000 years ago during the Auronian period. Unlike many later Paleolithic caves where images accumulated gradually over millennia, as generations added new paintings to existing panels, Chauvet appears to have been decorated during a relatively brief period, creating a remarkably coherent artistic program. The cave contains over 400 animal representations including horses, lions, rhinoceroses, bison, mammoths, and bears, rendered with extraordinary naturalism and technical sophistication. Artists employed advanced techniques, including careful preparation of cave surfaces, through scraping to create smooth painting substrates, use of the cave wall's natural contours to create three-dimensional effects, application of shading to suggest volume and musculature, and employment of perspective through overlapping figures to create depth. The presence of these sophisticated artistic techniques at the very beginning of the European Paleolithic art tradition suggests that the skills did not develop gradually within Europe but rather arrived fully formed with migrating populations who brought artistic knowledge developed elsewhere, possibly in Africa or Western Asia, where earlier traditions have left fewer preserved examples. Subject matter of Paleolithic cave art reveals consistent patterns across sites and time periods, with large herbivores dominating imagery while carnivores, humans, plants, and landscape features appear rarely. At Chauvet, dangerous animals including cave lions, cave bears, rhinoceroses, and mammoths appear frequently, an unusual pattern as most Paleolithic sites emphasize the prey species that people actually hunted and consumed, such as horses, bison, and reindeer. This focus on powerful, dangerous animals at Chauvet suggests the images served purposes beyond simple hunting magic or food documentation, possibly relating to spiritual beliefs about animal power, shamanic practices involving animal spirits, or narratives about creation and the relationship between humans and other beings. Hand stencils, created by placing a hand against the wall and blowing pigment around it, appear at many cave art sites, including some of the oldest, providing direct physical connection between modern viewers and Ice Age artists. The outlines preserve the exact size and shape of hands that touched these walls tens of thousands of years ago. At Maltravieso Cave in Spain, hand stencils created by Neanderthals at least 66,700 years ago demonstrate that symbolic marking of cave walls predates Homo sapiens in Europe, indicating that this behavioral capacity was shared by multiple hominin species or inherited from their common ancestor. Abstract signs and geometric patterns appear alongside animal imagery at most decorated caves, including dots, lines, grids, zigzags, and more complex patterns whose meanings remain enigmatic. At La Pasiega Cave in Spain, a red ladder-shaped symbol consisting of horizontal and vertical lines dates to at least 64,800 years ago, created by Neanderthals, and representing one of the oldest known abstract markings in cave art. These non-figurative images may represent proto-writing, notation systems tracking astronomical events or animal migrations, clan symbols marking territorial boundaries, or purely decorative elements without referential meaning. Some researchers have proposed that certain recurring abstract patterns might represent entoptic phenomena, geometric visual patterns generated within the eye and brain rather than through external stimuli, which people experience during altered states of consciousness induced by sensory deprivation, rhythmic drumming, or psychoactive substances, if correct. This interpretation would link cave art to shamanic practices where spiritual specialists entered trance states to communicate with supernatural realms, recording their visions on cave walls. The deep cave locations where much art appears, sometimes requiring crawling through narrow passages, wading through underground streams, or descending into chambers hundreds of meters from cave entrances, suggests that creating and viewing this art involved deliberate journeys into darkness, perhaps as part of ritual practices or initiations rather than casual aesthetic activities. The pigments used in cave paintings demonstrate sophisticated knowledge of mineral properties and chemical processes. Red ochre, derived from iron oxide minerals, appears most frequently, ranging from bright red to dark brown, depending on the specific mineral composition and heating treatment. Yellow ochre, chemically similar but in a different oxidation state, was also used, sometimes intentionally heated, to transform it into red ochre through controlled pyrolysis. Black pigments came from manganese dioxide minerals, or from charcoal created by burning wood or bone. White pigments, though rarer, were made from clay or calcite. Artists didn't simply crush these minerals and apply them directly, but rather process them through grinding, washing to remove impurities, mixing with binders like animal fat or plant resins to create paint that would adhere to rock surfaces, and sometimes heating to alter color properties. Analysis of paint residues from various cave art sites reveals that different caves use different recipes, suggesting that knowledge about pigment preparation was transmitted within cultural groups and that regional traditions developed distinct approaches. Some paintings show evidence of spraying technique, where liquid pigment was blown through hollow bones or reeds to create diffuse coverage or stenciled images. Other works were clearly painted with brushes made from animal hair, plant fibers, or feathers, while some appear to have been applied with fingers or pads of fur or moss. The distribution of cave art across the landscape reveals intentional site selection based on acoustic properties, visual impact, and accessibility. Researchers mapping cave art locations have discovered that painted chambers often possess unusual acoustic characteristics, including strong echoes, resonance at particular frequencies, and sound amplification properties. Some sites show correlation between image locations and acoustic sweet spots, where sound effects are most pronounced, suggesting that the visual experience of viewing art may have been combined with auditory experiences including chanting, singing, percussion, or the playing of bone flutes that would have produced dramatic sonic effects in these resonant chambers. The inaccessibility of many painted chambers, requiring difficult and sometimes dangerous journeys into the Earth's depths, implies that viewing this art was not a casual everyday activity, but rather reserved for special occasions, specific individuals, or particular life stages such as initiations. The effort required to reach these chambers, navigating through total darkness with only primitive lighting, from animal fat lamps or torches, would have created powerful psychological experiences of sensory deprivation, disorientation, and awe that likely heightened the emotional and spiritual impact of encountering the painted images. Beyond Europe and Indonesia, cave art and rock art traditions appear across Africa, Asia, Australia, and the Americas, demonstrating that visual artistic expression represents a universal human behavior rather than a regional cultural development. In Africa, the continent where Homo sapiens evolved, rock art traditions extend back at least 73,000 years as evidenced by the engraved ochre piece from Blombos cave in South Africa bearing a deliberate cross-hatched pattern, the oldest known drawing by a human. Australian Aboriginal rock art traditions represent some of the longest continuous cultural practices in human history, with the earliest dated examples reaching back approximately 40,000 years, and traditions continuing into the present day as living cultural practices rather than extinct archaeological phenomena. The Bradshaw paintings of Western Australia, also called Gwion art, depict human figures in elaborate costumes and headdresses engaged in various activities, though their age remains contested, with some researchers proposing antiquity exceeding 20,000 years, while others suggest younger ages. In the Americas, rock art traditions emerged soon after human colonization, with sites in Brazil and Argentina containing imagery potentially dating to 12,000 to 9,000 years ago, though dating rock art in the Americas remains technically challenging. The function and meaning of Ice Age cave art has generated extensive scholarly debate, with interpretations ranging from hunting magic to shamanistic ritual to art for art's sake. The hunting magic hypothesis, popular in the early 20th century, proposed that depicting animals on cave walls was believed to magically increase hunting success or give hunters power over prey species, a form of sympathetic magic where affecting an image affects the real animal it represents. This interpretation drew support from ethnographic accounts of hunting rituals among recent foraging societies, though critics noted that the animals most commonly depicted in caves don't always match the species most frequently hunted and consumed, as indicated by archaeological faunal remains. The structuralist interpretation, developed by André Leroy-Gourhan in the 1960s, proposed that cave art represented a symbolic system where different animal species corresponded to male and female principles, with their spatial arrangement within caves following consistent compositional rules, reflecting cosmological beliefs. While this approach moved beyond simplistic functionalism to recognize cave art as expressing complex symbolic systems, subsequent research found numerous exceptions to Leroy-Gourhan's proposed patterns, undermining claims of universal structural rules. More recent interpretations emphasize shamanistic practices, proposing that cave art was created by or for ritual specialists who entered altered states of consciousness to communicate with spiritual realms. According to this view, the animals, therianthropes, and abstract patterns depicted in caves represent visions experienced during trance states, rather than literal documentation of the physical world. The deep cave locations, requiring sensory deprivation and physical ordeal to reach, would have facilitated entry into altered states even without psychoactive substances, though some researchers propose that Ice Age peoples may have used naturally occurring hallucinogenic plants or fungi as part of ritual practices. The representation of therianthropes, beings combining human and animal features, appears consistent with shamanistic beliefs documented ethnographically, where spiritual practitioners report transforming into animals or encountering hybrid supernatural beings during trance journeys. However, the shamanistic interpretation faces criticism for projecting ethnographic patterns observed in recent societies onto ancient contexts where beliefs and practices may have been fundamentally different, and for assuming universal cognitive responses to altered states, rather than recognizing cultural specificity in how people interpret unusual experiences. Ultimately, the motivations behind Ice Age cave art likely varied across regions, time periods, and individual sites, encompassing multiple functions including ritual activity, narrative storytelling, territorial marking, education of youth, aesthetic expression, and purposes we cannot reconstruct from archaeological evidence alone. What remains certain is that by at least 50,000 years ago, humans across multiple continents possessed the cognitive sophistication, technical skill, and symbolic capacity to create enduring visual images that continue to move, inspire, and mystify viewers tens of thousands of years after their creation. The technological sophistication visible in Paleolithic cave art reveals that Ice Age peoples possessed detailed knowledge of animal anatomy, movement, and behavior that could only have been acquired through careful, sustained observation. The animals depicted at Chauvet and other caves are not schematic or stylized, but rather demonstrate naturalistic rendering of musculature, posture, facial features, and characteristic behaviors. Mammoths shown in profile with raised trunks, horses with heads lowered to graze, lions stalking prey with bodies low to the ground. Some paintings show animals in motion, with legs positioned to suggest running or walking, anticipating by millennia the development of techniques for depicting movement that would not be systematically explored again until much later in art history. The accuracy of these depictions has allowed paleontologists to identify extinct species and reconstruct details of their appearance and behavior that fossil bones alone cannot reveal, including coat patterns, ear positions, tail lengths, and social behaviors. This precision indicates that artists were not working from vague memories or creating generic representations, but rather drawing upon detailed mental models built through extensive direct experience observing these animals in their natural environments. The preservation of cave art depends on specific environmental conditions that have protected images for tens of thousands of years, while simultaneously limiting where art survives in the archaeological record. Deep caves with stable climates, minimal water infiltration, and protection from sunlight, wind, and temperature fluctuations provide ideal preservation environments, where pigments remain adhered to rock surfaces and calcite deposits seal images beneath protective mineral layers. However, this preservation bias means that art created in open-air rock shelters, on portable objects, or using organic materials like bark, hide, or wood has largely vanished, leaving only the tiny fraction that was created in deep caves or protected rock shelters. The surviving cave art therefore represents only a small sample of Ice Age visual culture, likely biased toward particular types of imagery, functions, and contexts, while entirely missing other artistic traditions that may have been equally or more important to prehistoric peoples. Rock art in exposed locations gradually fades due to weathering, lichen growth, and chemical dissolution, creating a temporal bias where older open-air art disappears while more recent images remain visible, complicating efforts to reconstruct the full chronological development of artistic traditions. Modern threats to cave art preservation include increased tourism that introduces moisture, carbon dioxide, and microorganisms into previously stable cave environments, causing accelerated degradation of pigments and rock surfaces. At Lascaux Cave in France, one of the most famous Paleolithic art sites discovered in 1940, visitor impacts caused such severe deterioration that the cave was closed to the public in 1963, with access now limited to researchers, while tourists visit a full-scale replica constructed nearby. Many other decorated caves face similar challenges, requiring difficult decisions about balancing public access against long-term preservation. Advanced digital documentation techniques including high-resolution photography, 3D laser scanning, and photogrammetry now allow researchers to create detailed virtual models of cave art sites, preserving a permanent digital record even as the physical originals continue deteriorating and enabling wider public access through virtual tours while protecting fragile original sites. These technologies also permit analyses impossible in the caves themselves, including precise measurement of image dimensions, examination of overlapping paintings to establish chronological sequences, identification of pigment compositions through spectroscopy without sampling, and reconstruction of how images would have appeared under flickering torchlight rather than modern electric illumination. The social organization required to produce cave art provides insights into Ice Age group structure, knowledge transmission, and cultural continuity. Creating sophisticated naturalistic imagery requires years of practice and skill development, implying that some individuals specialized in artistic production rather than everyone possessing equal artistic capacity. Whether these specialists were full-time artists supported by community provisions, part-time practitioners who also engaged in hunting and gathering, or ritual specialists whose artistic activities formed one component of broader spiritual roles remains unknown. The consistency of artistic conventions within regional traditions, particular ways of rendering horse manes, conventional positions for animal legs, characteristic color palettes, suggests that artistic knowledge was transmitted from experienced practitioners to novices through teaching and observation, rather than each artist independently inventing techniques. Some caves contain images clearly made by children, identifiable by their hand size in stencils and the height at which images appear on walls, indicating that young people participated in cave art activities, possibly as part of education or initiation practices. The presence of child handprints alongside adult prints in some caves creates poignant connections across time, preserving moments when families or groups, including children of various ages, made their marks together in these profound underground spaces. The legacy of Paleolithic cave art extends far beyond its archaeological significance to influence modern artistic, philosophical, and cultural consciousness. The discovery of cave art in the 19th and early 20th centuries fundamentally altered European understanding of prehistory, demonstrating that Ice Age peoples possessed aesthetic sensibilities, symbolic thinking, and technical skills comparable to modern humans, rather than being primitive savages as earlier evolutionary theories had assumed. Artists including Picasso, who visited cave art sites, drew inspiration from Paleolithic imagery, finding in these ancient works a directness, power, and essential quality that they felt modern art had lost. The existence of sophisticated art created tens of thousands of years ago by people living as mobile hunter-gatherers challenges assumptions about the relationship between artistic achievement and social complexity, demonstrating that profound symbolic expression does not require agricultural surplus, sedentary settlements, or hierarchical social organization. These images, created by people whose lifeways and belief systems we can only partially reconstruct, nevertheless communicate across vast expanses of time, preserving something essential about human creativity, imagination, and the drive to mark permanent records of inner experience onto the physical world. The hands outlined on cave walls, the animals captured in mid-stride, the enigmatic patterns whose meanings have been lost, all testify to the ancient origins of humanity's symbolic consciousness and our species' unique capacity to transform fleeting thoughts and visions into enduring material, forms that outlast their creators by hundreds of generations. The transition from mobile hunting and gathering to sedentary agriculture represents one of the most consequential transformations in human history, fundamentally altering human society, ecology, demography, health, and social organization in ways that continue to shape the contemporary world. This shift, often called the Neolithic Revolution, did not occur as a sudden transformation but rather unfolded gradually over thousands of years, beginning approximately 11,500 years ago in the Fertile Crescent, a region spanning from the eastern Mediterranean coast through modern Syria, Turkey, Iraq, and Iran, where wild ancestors of wheat, barley, lentils, and other crops grew naturally. The term revolution misleadingly implies rapid change, yet the process involved centuries of experimentation, partial adoption, abandonment, and reinvention as human populations gradually developed the knowledge, technology, and social organization necessary to sustain agricultural lifeways. Multiple independent centers of agricultural origin emerged across the globe: the Fertile Crescent, China, Mesoamerica, the Andes, Sub-Saharan Africa, and Eastern North America, each domesticating different suites of plants and animals adapted to local conditions, and each following unique trajectories from foraging to farming. This independent parallel development demonstrates that agriculture was not a one-time invention, diffusing outward from a single source, but rather a convergent cultural evolution occurring when particular environmental, demographic, and social conditions aligned. The earliest evidence for deliberate plant cultivation comes from sites in the Levantine corridor and surrounding regions of Southwest Asia, where people began manipulating wild plant populations between 11,500 and 10,000 years ago. These early cultivators did not immediately abandon hunting and gathering, but rather incorporated cultivation as one strategy within broader subsistence systems that continue to include wild resource exploitation. Archaeological sites from this transitional period preserve seeds, grinding stones, storage pits, and other evidence indicating people were collecting, processing, and storing large quantities of wild cereal grains, including einkorn wheat, emmer wheat, and barley, activities that laid the foundation for later domestication. True domestication, the genetic modification of plants through human selection producing populations dependent on human intervention for reproduction, appears archaeologically around 10,500 to 10,000 years ago, when morphological changes in seed size, seed coat thickness, and rachis brittleness indicate that plants were being systematically selected for traits favoring human harvesting and consumption. Wild cereals have brittle rachises, the attachment points where seeds connect to seed heads, that shatter readily when ripe, dispersing seeds widely for natural propagation. Domesticated cereals have tough rachises that retain seeds on the plant, making harvesting easier but preventing natural seed dispersal and requiring humans to thresh and plant seeds intentionally. This mutation, while maladaptive in wild populations, became fixed in cultivated populations because humans preferentially harvested plants with non-shattering rachises and inadvertently planted their seeds year after year. Animal domestication followed similar patterns of gradual transformation from exploitation of wild populations to genetic modification through human-directed breeding. The earliest confirmed animal domestication involves dogs, which were derived from grey wolves through selection for tameness, trainability, and cooperative behavior, with genetic evidence suggesting this process began as early as 40,000 to 15,000 years ago in Eurasia. However, the domestication of food animals occurred considerably later, beginning around 10,500 years ago in Southwest Asia with sheep and goats, followed by cattle around 10,000 years ago, and pigs around 9,000 years ago. These animals were not domesticated simultaneously or in the same locations. Goats appear to have been first domesticated in the Zagros Mountains of Iran, sheep in eastern Anatolia, cattle possibly in multiple locations including the Fertile Crescent and the Indian subcontinent, and pigs independently in both the Near East and China. Domestication involved selecting for behavioral traits, including reduced flight response to humans, tolerance of confinement and social subordination, as well as morphological traits like smaller body size, shortened snouts, floppy ears, and color pattern variations that would be maladaptive in wild populations. These changes didn't require conscious understanding of genetics, but rather resulted from humans consistently choosing to breed the tamest, most manageable individuals generation after generation, inadvertently selecting for juvenile characteristics that persisted into adulthood, a phenomenon called neoteny. The motivations driving the shift from foraging to farming remain debated, with multiple interacting factors likely contributing to the transition. Population pressure represents one frequently cited driver. As human populations grew following the end of the Pleistocene, wild food resources in some regions may have become insufficient to support increasing numbers, creating incentives to augment natural productivity through deliberate cultivation and animal management. Climate change at the Pleistocene-Holocene boundary created new opportunities and pressures. Warming temperatures, increased precipitation, and elevated atmospheric CO2 concentrations enhanced plant productivity in some regions, while destabilizing ecosystems that hunter-gatherers had depended upon for millennia. The extinction of Pleistocene megafauna eliminated major prey species and forced subsistence reorientation towards smaller animals and increased reliance on plant foods. Social competition provides another potential motivation. In some societies, individuals or groups may have adopted agricultural intensification to produce surplus food that could be exchanged for prestige, political influence, or access to mates, initiating competitive dynamics where populations were drawn into farming even if it didn't improve individual welfare. Religious or ritual factors may have played roles. Deliberately planting, tending, and harvesting crops could have held symbolic or ceremonial significance that motivated agricultural practices before they became economically necessary. The consequences of agricultural adoption were profound and multifaceted, involving trade-offs rather than unambiguous improvements in human welfare. Agriculture enabled population growth by allowing higher population densities per unit area than hunting and gathering could support. Agricultural villages of hundreds or even thousands of people occupied territories that previously sustained only dozens of foragers. This population growth represented both cause and consequence of agriculture, as larger populations required more food production, which required more labor, which was provided by additional children, creating a self-reinforcing cycle. However, agricultural populations experienced elevated disease burdens compared to foragers, Sedentary living allowed accumulation of human waste, creating unsanitary conditions. Close proximity to domestic animals facilitated zoonotic disease transmission. Larger population sizes enabled crowd diseases like measles and smallpox to persist, and dietary dependence on limited staple crops caused nutritional deficiencies. Skeletal evidence indicates that early agricultural populations exhibited shorter stature, higher rates of dental disease, increased prevalence of infectious disease markers, and greater evidence of nutritional stress compared to contemporary hunter-gatherers. Life expectancy may have actually declined during the initial agricultural transition, before eventually recovering as farming techniques improved. Yet despite these costs, agriculture proved irreversible. Once populations grew beyond carrying capacity for hunting and gathering, returning to foraging became impossible, even if farming was arduous and unhealthy. The spread of agricultural lifeways from centers of origin to surrounding regions occurred through two primary mechanisms: endemic diffusion, where farming populations physically migrated into new territories, and cultural diffusion, where foraging populations adopted agricultural practices through observation, trade contact, or intermarriage with farmers. In Europe, genetic evidence indicates that agriculture spread primarily through endemic diffusion, as farming populations from Anatolia moved westward and northward, gradually replacing or absorbing resident hunter-gatherer populations over several thousand years. In sub-Saharan Africa, the Bantu expansion beginning around 3,000 years ago involved agricultural populations moving from West Africa southward and eastward across the continent, carrying with them domesticated crops including yams, sorghum, and millet, as well as iron-working technology and Bantu languages that now dominate across much of Africa. In the Americas, agricultural diffusion followed complex patterns, with maize agriculture spreading from Mesoamerica northward and southward over millennia, reaching the southwestern United States by 2,000 years ago and the eastern woodlands by 1,000 years ago, with local populations selectively adopting crops while maintaining hunting and gathering practices in a mixed economy. By 5,000 years ago, agricultural or pastoral economies had become the dominant mode of human subsistence across much of Eurasia, Africa, and the Americas, with hunter-gatherer populations increasingly confined to marginal environments unsuitable for agriculture, including deserts, Arctic tundra, tropical rainforests, and isolated islands. This agricultural transformation set the stage for all subsequent developments in human history, including urbanism, state formation, writing, specialized craft production, and eventually industrialization, making the Neolithic Revolution the pivot point between the Paleolithic world of mobile foraging and the modern world of settled agricultural and industrial civilization. Beyond the grand narratives of human evolution, migration, and agricultural revolution, prehistoric peoples developed countless innovations that addressed practical challenges of daily existence, gradually accumulating technological knowledge that transformed human capabilities and quality of life. The invention of pottery, approximately 20,000 years ago in East Asia, represents one of the earliest major technological achievements of the late Paleolithic period, fundamentally expanding food processing and storage possibilities. The earliest known pottery vessels come from sites in China and Japan, where hunter-gatherer populations created ceramic containers through deliberately shaping clay, allowing it to dry, and firing it at temperatures exceeding 500 degrees Celsius to transform soft clay into hard, durable, waterproof ceramic. This pyrotechnology required understanding that certain clay types become permanently hardened through heating, knowledge of how to construct and maintain fires hot enough for effective firing, and techniques for shaping clay into desired forms. Pottery enabled cooking methods impossible with organic containers. Foods could be boiled, stewed, or slowly simmered, making tough plant materials edible, rendering bones into nutritious broths, and detoxifying certain otherwise poisonous plants through prolonged cooking. Ceramic vessels also revolutionized food storage, allowing preservation of surplus production across seasons and providing sanitary, pest-resistant containers superior to baskets, leather bags, or wooden bowls. The creation of the first baked bread approximately 14,000 years ago at Shubaika 1, an archaeological site in northeastern Jordan, predates the advent of agriculture by several thousand years and demonstrates that complex food processing techniques emerged among hunter-gatherers before plant domestication. Charred bread-like remains recovered from hearths at Shubaika contain flour made from wild barley, wheat and oats, mixed with tubers from aquatic plants, shaped into flat cakes and baked on heated stones. The effort required to process wild cereals, collecting, threshing, winnowing, grinding into flour, mixing with water, and baking, significantly exceeds the labor needed to simply roast or boil whole grains, indicating that bread was valued sufficiently to justify this additional investment. The appeal of bread may have provided motivation for subsequent agricultural development, as people sought reliable access to cereal grains for bread production and thereby initiated the cultivation practices that eventually led to full domestication. Grinding stones, which become common at archaeological sites throughout the Near East and Eastern Mediterranean by 15,000 years ago, preserve wear patterns and starch residues, documenting intensive processing of wild cereals and other plant materials into flour, indicating that grinding and baking technology preceded and may have facilitated the transition to agriculture. Mining represents another profound technological threshold, extending human resource extraction beyond surface collection to deliberate excavation of underground materials. The earliest definitive evidence for mining comes from Swaziland in southern Africa, where ochre mines at Lion Cave date to approximately 43,000 years ago, with some researchers proposing dates potentially reaching 100,000 years ago, based on geological and archaeological associations. Prehistoric miners excavated shafts and galleries pursuing underground ochre deposits, removing overlying rock and soil to access prized pigment sources. The investment required for mining, locating subsurface deposits, removing large quantities of rock, shoring up unstable passages, providing lighting for underground work, demonstrates that ochre held exceptional value, far exceeding its practical utility as pigment and suggesting symbolic or ritual significance that justified extraordinary effort. Flint mining emerged later, with definitive evidence by 8,500 years ago from sites including Grimes Graves in England and Spienne in Belgium, where Neolithic peoples excavated extensive underground galleries, pursuing high-quality flint for manufacturing polished stone axes, blades, and other tools. These mines involved sophisticated engineering including vertical shafts exceeding 10 meters deep, radiating horizontal galleries following flint seams, wooden supports preventing collapse, and chalk or antler picks for excavation. Infrastructure that required coordinated labor, specialized knowledge, and long-term planning extending far beyond immediate subsistence needs. Textile production, though rarely preserved archaeologically due to the perishability of organic fibers, represents a crucial prehistoric innovation with profound implications for human ecology and society. The earliest definitive evidence for woven textiles comes from impressions preserved on clay objects dating to approximately 27,000 years ago, though indirect evidence, including bone needles, spindle whorls, and loom weights, suggests textile production may extend much earlier. Clothing manufacture involves multiple distinct technologies, including fiber production, through harvesting plant materials, or animal hair, fiber processing through cleaning and preparing materials for spinning, spinning to create thread by twisting fibers together, and weaving or sewing to create finished textiles. Each step requires specialized knowledge, tools, and motor skills transmitted through social learning across generations. The development of tailored, fitted clothing, rather than simple draped hides, enabled human occupation of extremely cold environments, including Ice Age Europe and eventually the Arctic, where survival would be impossible without effective insulation. Clothing also serves social functions including modesty, status display, group identification, and individual expression. Functions that may have been equally or more important than thermal protection in motivating textile production and innovation. The control of fire, discussed earlier in the context of its initial adoption, underwent continuous technological refinement as humans developed increasingly sophisticated methods for fire. The control of fire, discussed earlier in the context of its initial adoption, underwent continuous technological refinement as humans developed increasingly sophisticated methods for fire production, maintenance, and application. By the late Paleolithic, humans had mastered multiple techniques for generating fire on demand, rather than relying on capturing and maintaining flames from natural sources. Friction methods involved rapidly rotating a wooden drill against a wooden base, generating sufficient heat through friction to ignite tinder made from dried plant material, fungus, or other easily combustible substances. Percussion methods involved striking iron pyrite or similar minerals against flint, producing sparks that could ignite prepared tinder. These fire-starting technologies required not only mechanical skill, but also knowledge of material properties, which woods worked best for friction firemaking, which stones produced the hottest sparks, which tinders ignited most readily. Accumulated through generations of experimentation and transmitted through cultural learning. The development of hafting technology, attaching stone points, blades, or other tools to wooden or bone handles using adhesives, bindings, or both, represented a crucial innovation that dramatically improved tool effectiveness. Hafted tools provided mechanical advantages through leverage, allowed replacement of dulled or broken working edges without discarding entire implements, and enabled creation of composite tools, combining materials with complementary properties. Archaeological evidence for hafting extends back at least 300,000 years, with wear patterns on stone tools and preserved residues indicating attachment to handles. However, sophisticated hafting using complex adhesives became particularly important during the Middle Stone Age in Africa, where people manufactured birch bark tar, plant resins mixed with ochre, and other compounds specifically designed for securing stone points to shafts. These adhesives required multi-step production processes, including controlled heating, mixing of ingredients in specific proportions, and sometimes chemical transformation of raw materials, demonstrating that prehistoric peoples possessed practical chemistry knowledge applied systematically to solve engineering problems. Fishing technology evolved from simple hand catching and spearing to sophisticated systems including hooks, nets, weirs, and traps that allowed systematic exploitation of aquatic resources. Bonefish hooks appear in the archaeological record by at least 23,000 years ago, with some researchers proposing earlier origins based on indirect evidence. Net making required development of cordage technology, twisting plant fibers or animal sinews into strong, flexible string, then knotting these strings into mesh patterns suitable for different fishing contexts. Stone net sinkers and bone needles for net repair appeared at sites across Europe, Asia, and Africa by the late Paleolithic, indicating that net fishing had become an established practice. Weirs and fish traps constructed from wooden stakes, stone walls, or woven materials channeled fish into confined areas where they could be easily captured, requiring understanding of fish behavior, tidal patterns or seasonal migrations, and hydrology. These fishing technologies enabled populations to incorporate abundant, reliable aquatic protein into diets, reducing dependence on terrestrial hunting and supporting larger, more sedentary populations along coastlines, rivers, and lakes. The everyday life of prehistoric peoples involved constant awareness of seasonal cycles, detailed knowledge of plant and animal ecology, and sophisticated spatial cognition, enabling navigation across extensive territories without maps or written records. Hunter-gatherers maintained mental maps encoding locations of water sources, food plants, game animal territories, stone tool raw materials, medicinal plants, and hazards including predators and hostile human groups. This environmental knowledge wasn't innate, but rather learned through years of experience, observation, and instruction from knowledgeable elders who transmitted ecological information across generations. Seasonal rounds involved planned movements between different resource patches as plants ripened, animals migrated, or water availability changed, requiring advanced planning and coordination among group members. Archaeological sites preserve evidence of these movements through seasonal indicators, including birthing patterns in hunted animals, ripening times of consumed plant species, and migrations of fish or waterfowl exploited at particular locations. Social organization in prehistoric societies likely involved flexible, egalitarian band-level groups of 25 to 50 individuals connected through kinship, marriage, and alliance networks to other bands within regional populations, numbering hundreds or thousands. These small groups maximized mobility, facilitated resource sharing, and maintained personal familiarity among all members, yet they weren't isolated. Periodic aggregations for trade, mate exchange, ritual activities, and information sharing connected bands within larger social networks. Evidence for long-distance exchange networks includes marine shells found hundreds of kilometers inland, stone tools made from raw materials available only in distant locations, and stylistic similarities in ornaments or art across broad geographic regions, all indicating that prehistoric peoples maintained social connections spanning vast distances, despite the absence of permanent settlements or centralized political authority. The innovations developed during prehistory, from fire control to pottery to composite tools, weren't merely technical achievements, but rather emerged from and reinforced complex social systems where knowledge transmission, cooperation, and cultural learning enabled human populations to occupy diverse environments and adapt to changing conditions through accumulated wisdom rather than purely instinctive responses. The boundary between prehistory and history remains fundamentally arbitrary, defined conventionally as the advent of writing systems that allow direct access to ancient people's own accounts of their lives, beliefs, and events, rather than relying exclusively on archaeological interpretation of material remains. This definition places the end of prehistory at different times in different regions, depending on when literate civilizations developed, or when non-literate societies came into contact with literate neighbors who recorded information about them. In Mesopotamia and Egypt, writing emerged approximately 5,600 to 5,000 years ago, making these among the earliest regions to transition into history. In China, writing developed around 3,200 years ago during the Shang dynasty. The Indus Valley civilization may have possessed writing by 4,500 years ago, though the script remains undeciphered, leaving uncertainty about whether these symbols represent true writing or non-linguistic notation. Mesoamerican civilizations, including the Maya, developed writing systems approximately 2,400 years ago. For many regions, including sub-Saharan Africa, northern Europe, the Pacific Islands, and the Americas north of Mexico, prehistory extended into the past few centuries when European colonization brought literate observers who created written records describing indigenous peoples. The development of writing represented not a sudden invention, but rather a gradual evolution, from simpler notation systems through increasingly complex symbolic representation toward full linguistic encoding. The earliest proto-writing consists of tokens, small clay objects in various shapes representing commodities like grain, livestock, or oil, used in the Near East by at least 8,000 years ago for record-keeping in early agricultural economies. These three-dimensional tokens evolved into two-dimensional pictographic symbols, impressed or drawn on clay tablets, initially representing concrete objects, but gradually acquiring capacity to denote abstract concepts, actions, and grammatical elements. Cuneiform script in Mesopotamia and hieroglyphic writing in Egypt both emerged from these pictographic origins, developing rebus principles where symbols could represent sounds rather than only meanings, allowing representation of any word in the spoken language rather than only those concepts for which pictographic symbols existed. This phonetic breakthrough transformed writing from limited accounting notation into a flexible system capable of recording laws, literature, historical narratives, religious texts, and the full range of human expression. The motivations driving writing's development were primarily administrative and economic, rather than literary or historical. Early texts overwhelmingly consist of economic records documenting transactions, inventories, tax payments, and resource allocations within increasingly complex agricultural societies, where oral memory proved insufficient for tracking obligations and exchanges. Writing enabled state formation by allowing centralized authorities to monitor resources, levy taxes, organize labor for public works, and maintain bureaucratic control over large populations across extensive territories. Religious institutions also drove literacy development, as priests recorded ritual procedures, maintained calendars tracking agricultural cycles and astronomical events, and preserved sacred narratives. Only later did writing expand to include legal codes, royal propaganda, historical chronicles, and eventually literature created for aesthetic or entertainment purposes. The social distribution of literacy remained highly restricted. In most early civilizations, only specialized scribes trained through years of instruction could read and write, with the vast majority of the population remaining illiterate throughout their lives. The transition from prehistory to history didn't mark a fundamental cognitive or cultural transformation, but rather represented a technological innovation that changed how cultural information was stored and transmitted. Prehistoric peoples possessed the same mental capabilities, emotional lives, social complexity, and creative potential as historical and modern humans. The absence of writing doesn't indicate absence of sophisticated culture, only absence of direct written testimony. The archaeological record preserves abundant evidence for prehistoric symbolic thought, artistic expression, technological innovation, and social organization that in many respects equals or exceeds the achievements of early literate civilizations. The stunning cave paintings of Chauvet, created 32,000 years ago, demonstrate technical and artistic sophistication matching works produced by literate societies tens of thousands of years later. The architectural achievements of Neolithic societies, including megalithic structures like Stonehenge constructed without writing, metallurgy, or wheeled vehicles, testify to sophisticated engineering knowledge and social coordination. The successful colonization of every habitable continent by mobile hunter-gatherers possessing only oral culture reveals adaptive capacity and behavioral flexibility that written records alone could not have improved. The legacy of prehistory extends far beyond its chronological span, fundamentally shaping the biological, cultural, and ecological foundations upon which all subsequent human history has been built. The human body evolved its current form during the Pleistocene, with anatomical features, physiological systems, and cognitive architecture reflecting adaptations to hunting-gathering lifeways in African and Eurasian environments, rather than to agricultural or industrial conditions. This evolutionary mismatch between the body's evolution shaped and the environments modern humans inhabit helps explain numerous health challenges, including obesity, type 2 diabetes, cardiovascular disease, and psychological disorders that emerge from conflicts between prehistoric adaptations and contemporary conditions. Human nutritional requirements, physical activity patterns optimal for health, social bonding mechanisms, and stress responses all evolved in contexts drastically different from modern urban industrial environments, creating tensions between biological heritage and cultural reality. The genetic diversity observable in contemporary human populations largely accumulated during prehistory, with the major population divergences, migration events, and admixture episodes that created modern geographic variation in physical traits, disease susceptibilities, and genetic adaptations all occurring before the advent of writing. The reduced genetic diversity in non-African populations relative to African populations reflects the out-of-Africa bottleneck approximately 60,000 years ago, while the presence of Neanderthal and Denisovan DNA in modern genomes documents prehistoric interbreeding between divergent hominin lineages. These ancient admixture events continue influencing contemporary health, with archaic human alleles affecting immune function, metabolic efficiency, high-altitude adaptation, and numerous other phenotypic traits in present-day populations. The population structure observable today, the clustering of genetic variation into continental groups, the clinal patterns of trait frequency change across geographic space, the correlation between genetic and linguistic relationships, all emerged during prehistoric dispersals and isolation by distance processes operating over thousands of generations. Cultural diversity, though continuously evolving and transforming, retains deep connections to prehistoric roots visible in linguistic relationships, subsistence patterns, kinship systems, and religious beliefs. The major language families, Indo-European, Sino-Tibetan, Afro-Asiatic, Niger-Congo, Austronesian, and others, emerged during prehistory through population dispersals and language diversification processes that began thousands of years before writing recorded any linguistic data. Reconstructing these ancient languages through comparative methods reveals vocabulary reflecting prehistoric technologies, environments, and social organization. Proto-Indo-European terms for wheeled vehicles, domesticated animals, and patrilineal kinship reflect the pastoral societies of the Pontic-Caspian steppe 5,000 to 6,000 years ago, while Austronesian vocabulary for canoe construction, navigation, and oceanic resources documents the maritime adaptation that enabled prehistoric colonization of Pacific islands. Religious beliefs across diverse cultures retain elements traceable to prehistoric shamanism, animism, and ancestor veneration, suggesting deep temporal continuity in how humans conceive of supernatural forces, spiritual realms, and the relationship between living and dead. The ecological transformations initiated during prehistory, megafaunal extinctions, anthropogenic fire regimes, vegetation modifications, and ultimately, agricultural landscape conversion, profoundly altered Earth's ecosystems in ways that persist and intensify in the present. The loss of Pleistocene megafauna removed ecological functions that smaller surviving animals cannot replicate, fundamentally restructuring nutrient cycling, vegetation dynamics, seed dispersal, and trophic interactions across terrestrial ecosystems worldwide. Contemporary rewilding initiatives attempting to restore ecosystem functions through reintroduction of large herbivores represent efforts to partially reverse prehistoric megafaunal losses, recognizing that modern landscapes are impoverished remnants of prehistorically richer biological communities. The adoption of agriculture approximately 11,500 years ago initiated a trajectory of landscape transformation that has accelerated through history, with current estimates suggesting that humans have significantly modified approximately 75% of Earth's ice-free land surface primarily through agricultural conversion, a process that began in prehistory and continues expanding. The technological and conceptual foundations for contemporary civilization, including controlled energy use, material processing, tool manufacture, symbolic communication, social cooperation, and environmental manipulation, all emerged during prehistory through cumulative cultural evolution operating over thousands of generations. Fire control, first achieved approximately 400,000 years ago, represents humanity's earliest systematic energy technology, enabling cooking, heating, manufacturing, and landscape modification that foreshadowed the fossil fuel energy systems driving modern industrial civilization. Stone tool manufacture, practiced for 3.3 million years, established principles of raw material selection, manufacturing sequences, quality control, and tool design that underlie all subsequent material technologies, from ceramics to metallurgy to synthetic polymers. The development of composite tools combining materials with complementary properties, stone points hafted to wooden shafts using adhesives, anticipated the engineering logic of modern composite materials and modular design. Pyrotechnology applied to pottery production around 20,000 years ago established technical knowledge about high-temperature material transformations that later enabled metallurgy, allowing copper smelting by 7,000 years ago, bronze casting by 5,000 years ago, and iron working by 3,200 years ago. each advance building on accumulated prehistoric knowledge about fire control and material properties. The cognitive capabilities that enable modern science, technology, and culture, including abstract reasoning, causal understanding, hypothesis testing, social learning, and symbolic representation, evolved during prehistory and are visible archaeologically in the sophisticated problem-solving, innovation, and cultural transmission documented throughout the Paleolithic and Neolithic periods. The capacity for symbolic thought, manifested in cave art, personal ornaments, burial practices, and abstract notation, emerged at least 100,000 years ago and possibly earlier, providing the cognitive foundation for all subsequent developments in language, art, religion, mathematics, and science. The human propensity for cumulative cultural evolution, where innovations are retained, recombined, and incrementally improved across generations, rather than repeatedly rediscovered, enabled prehistoric populations to accumulate technological and ecological knowledge far exceeding what any individual could acquire through personal experience alone. This cultural learning capacity, more than any specific invention, represents prehistory's most profound legacy, enabling human populations to occupy diverse environments, adapt to changing conditions, and eventually transform from one species among millions into the dominant force shaping Earth's biosphere. Understanding prehistory remains essential for comprehending the human condition in the modern world. Our evolutionary heritage, our biological capabilities and limitations, our psychological tendencies and social needs, our relationship with the natural environment, and the deep historical processes that have shaped contemporary diversity and inequality. The archaeological record documents that for the vast majority of human existence, over 95% of the time since our species emerged, people lived as mobile hunter-gatherers in small egalitarian bands, subsisting on wild resources without permanent settlements, complex hierarchies, or intensive environmental modification. Agriculture, cities, states, writing, and all the familiar trappings of civilization represent recent innovations overlaying this deeper hunter-gatherer heritage that shaped human biology and psychology. Many contemporary social problems, including conflicts over resources, hierarchical dominance, intergroup violence, and environmental degradation, have prehistoric roots visible in the archaeological record, demonstrating that these challenges aren't recent inventions, but rather long-standing human tendencies that cultural institutions must work to constrain. Prehistory also offers hope and inspiration, documenting human resilience, creativity, and adaptability across millennia of environmental change, population movements, and cultural transformations. Prehistoric peoples survived the last glacial maximum when ice sheets covered much of the northern hemisphere, adapted to rapidly changing climates during the Pleistocene-Holocene transition, colonized environments ranging from Arctic tundra to tropical rainforests to high mountain plateaus, developed hundreds of distinct cultural traditions and linguistic systems, and created art, music, and symbolic expressions that continue to move contemporary viewers. These achievements were accomplished with relatively simple technologies but sophisticated knowledge, extensive cooperation, and remarkable behavioral flexibility, qualities that remain essential as humanity confronts contemporary challenges including climate change, biodiversity loss, resource depletion, and pandemic disease. The millions of years of human prehistory that preceded writing's invention just 5,600 years ago shaped who we are biologically, cognitively, and culturally in ways that cannot be erased or ignored, forming the deep foundation upon which all subsequent history has been built and upon which the human future, whatever form it takes, will continue to rest.